departure in case.
Okay, yes, thank you.
Text to run this number.
Yes, I think I got it.
Yeah.
Yes, thank you.
There's going to be a one is remembers GIAN system.
Okay.
This is reader memory.
Mhm. at the beginning, we'll have a short lecture about neurodegeneration models, then about drug development in general. Then we have exercise type seminar, and after that it depends how much time will be left. We will have a lecture about the mitochondrial role and mitochondrial role in neurodegeneration as well. So first about models. Very generally, what models are in use, then more specifically about the animal models, and then as an example what kind of models we could use if we specifically want to model the early onset Parkinson's disease. So the first part will be generic. So we have very different kind of in vitro and in vivo models available. So it starts from the cell lines, then next step is neuronal cell lines, then primary cells, in our case neurons, then patient-derived IPSIs, which is differentiated into neurons, and co-cultures, and easier or simpler in vivo models, non-mammalian ones, and then mammalian rodent models. So it's most easy to use simple cell lines, HEC, HeLa, what people are using. So the advantage is that it's easy and it is cheap and it is fast. But you can do only very limited things with these cell lines because they really lack most of the relevant components that neurons have. So they don't replicate really what happens in neurons. So it's still good if you study some kind of basic mechanism, then the basic mechanism is not neuron specific, then you can study it. If your work is only, if you study neurodegenerative diseases and your work is only with these simple cell lines, then you have difficulties to publish well. You can't really do any translational work only with these simple cell lines. So the next step that helps a little bit is to use neuronal cell lines, PC6, PC12, rat, chromocytoma cells, or human neuroblastoma cells, this SH, SAF5, or neuro2 A cells. So they are slightly better because they have some neuron-like features. So you see some neurids growing there. But they are not fully matured, they do not have synapses, so they don't form the network between the cells. But they are already slightly better, so if you can choose whether to use these simple cell lines or neuronal cell lines, then these neuronal cell lines are slightly better to use. But they are already more difficult to maintain. For example, if you take PC cells, they are not growing so fast. The coating of the dishes is, if you use glass bottom dishes, is more expensive. And if you do microscopy, then it's okay, but if you do the biochemistry, then you have less material, everything is slower. So then the next level is the primary cells, neurons. So we use mostly primary neurons. So the primary neurons we will get them from the rat pups. So we will take 1 day old rat pups and we will prepare the culture from the cortex. So we have a cortical neurons. It's the easiest way because cortex is big, we will get a lot of cells. It's not enough for biochemistry, but it's enough for imaging. You can also have different kind of neurons. You can have a cerebellar granular cells, you can have a dorsal root ganglion cells, but since these brain areas, cerebellum is big, but other areas is relatively small, you don't get too many neurons. But again, if you do the imaging, then you don't need so much neurons. And they are real neurons, so if you wait a week, then you have a nice axons. If you wait more than 11 days, then you will have already the synapses. You can say that they are almost mature. And if you want to have a really mature neurons, then it's day 70, 80 or something after you plate them. Another thing is how you will model exactly your disease in these cells. So for that we have 2 possibilities. One is we have, in case of cell lines, we can have a cell line which is expressing something. It's, for example, some kind of synuclein, beta amyloid. It's additional gene is introduced, those genes are introduced, and you have increased expression of that. Or something is knocked out in these cell lines. For example, you don't have a pink or Parkin expressed there at all. Or something is knocked in, the mutation is knocked into this gene. So you can do that with cell lines. You can't do that so easily with neurons, because neurons you will prepare, then you will use them a week or 2 later. You can't really do that way. You can't have a cell line expressing something. Or so in that case we transfect the neurons with plasmids, or we will infect them with viral vectors expressing something which is leading to accumulation or so, or we are treating the neurons with siRNA or transfecting with siRNA encoding plasmids to knock something down. So it's also possible to use CRISP system in this culture to knock something in or out. This is slightly more complicated, but doable as well. So the result you get is much more closer to real life, but the complexity is much higher. So to prepare the neurons, you need rat pups to do the work, to everything, to transfect, to image. It is already much more expensive. You need special media, neurobasal media components, P27, they are already expensive. But it is, what you get out is valuable. So you can publish, if you use such kind of primary cells, then you can already publish without problems in good journals. What we are usually doing, we are taking the normal cells and then we are knocking something down or overexpressing something. Another option is to prepare these cells from transgenic mice. It's also doable, but it is much more complicated and much more expensive. So then next level is to have the human neurons coming from the human or patient-derived IPSIs. So IPSIs are these induced pluripotent stem cells. So how it works, mostly if you have a patient, you will take the fibroblasts from the patient, then you will purify these IPSIs from the fibroblasts, and then you reprogram them to get your neurons. So you can get, if you do it properly, you can get even in dopaminergic neurons. But this is really, really expensive and very time consuming. So if you want to do, it depends what you want to do. If you want to study the basic mechanism related to some neurodegenerative diseases, and you have to do a lot of manipulation to introduce new genes, to suppress something, then you can do that with these patient-derived IPSIs and neurons from them. It's too complicated. So this is just to prove that something which is already discovered, that this mechanism is relevant and affecting the neurons. So that way you will just control it in these IPSIs. So if you take papers, then they not always, but quite frequently start with simple system, neurons, neuronal cell lines, and then they will just, check, is that also this phenomena present in these patient-derived neurons. Why these patient-derived neurons are most important? First, they are human, and second, they have the genetic background of the patient. If you take the mice model or rat model, rat neurons, then the genetic background is rat mice genetic background, it's similar everywhere. But in the case of patients, the background, this means other genes, other mutations, they are all different. They are affecting how this disease is progressing. This is one reason why the disease could progress completely differently in these human neurons than in rat neurons. So this is just what we are planning to use it, and using its final check whether our earlier discoveries were correct and whether the same is happening in human neurons. But it's time consuming. If we are doing this primary neuron experiment, then we prepare the cells, for example, in our lab today. We transfect them Friday, and then next Friday or 2 weeks later, if we want to have a lot of synapses and mature neurons, we will do the experiments. So the cycle is 2 weeks, from maximum 2 weeks from rat pup to data. If we will do this, then usually you have to prepare the IPSIs, but then you can store them. But the reprogramming itself is taking already a month. So from the frozen IPS to neuron, you will have to spend 1 month at least. And also, since it is you need statistics, in this case you have in one culture you have all controls and treatments. Here you need different cultures. You need a control culture, patient culture, or free controls, free patient cultures, or another option is to have one patient and mutations corrected. Even in that case, it's 3 cultures together. So it's much more problematic, but helpful. Then one level up is co-cultures and organotypic cultures. So when I had lectures yesterday and before that, then you saw that quite frequently it's not just neuron that is doing something. It is how neuron is interacting with glia, astrocytes and other cells. So if you are just having the neuronal culture, then it's sometimes not enough to see or to understand what really happens. There is constant interaction between glia and neurons. So to understand that, you need co-culture. You need different kind of cells. if you prepare the cortical culture, it's of course not just neuronal culture, it's a mix of everything, but they are in different, so to say, places. Co-culture may mean that you have one layer of the glial cells and another layer of the neurons, so it's sometimes helping. And organotypic cultures is that you are forming some kind of organoids, spheroids, so where all these cells are growing together, and this is even closer to the brain. But again, it's one step more complex. You can't really study the mechanism there anymore. In most of cases, it is just to do some checks and to study something very specific. It's not having any throughput. Then if you study disease, if you study some basic mechanism, then it's okay. But if you study some kind of mechanism and you will say that, this is relevant in case of neurodegeneration, or you study some drugs, you want to say that is helping in case of neurodegeneration, then the journals are expecting to have some kind of in vivo model. So it's basic journals, they are not so nasty, but if it's more clinical journals, or semi-clinical journals, they sometimes say that in vivo data is necessary, and without that we don't consider your paper at all. So if you are very close to disease. So the easiest way, which is also a little bit helpful, is to use non-mammalian models. Zebrafish, Drosophila, and less C. elegans. So what is good? Again, it's relatively easy. Drosophila is very easy to study, zebrafish slightly less easy, but also okay, it's at least bigger. And they are good to study whether your treatment is effective, whether your drug is really helping, if you can model it somehow in zebrafish or Drosophila, the disease. And they are also good for screening. So you can't screen hundreds or thousands of compounds in zebrafish screen, but 10 to 20 is still okay. So this is the way to do such kind of first in vivo test. But what is usually expected is to have mammalian, usually rodent in vivo model. So there are two kinds of mammalian models, if you can divide them like that. One is that you have a chemically induced disease model. Means you have compound or pharmacological agent, and this you are using this to have a disease. Usually this is not so good. This is what people did before transgenic animals came. So nowadays the standard is to use genetic models of mice and rats mostly that express or do not express human disease-related genes. Why these transgenic models are best? They are really reflecting what happens in brain. All these aspects of disease are very similar to human disease, and you can do what is most important. You can do histology and behavioral analysis. So if you have a motor symptoms in human, then you can check whether the same motor symptoms are present in rats or mice. And if you treat with something, you can see whether the symptoms are going away. It's still not super, it means not fully similar to humans, and the problem is the cost. So in our countries the animal houses are very expensive, So if I want to do some kind of 1 year study with 100 rats, treated every day with something, then it will cost me maybe at the end 100,000 euros. So that's really big money, so we can't do it so easily. Okay, then to summarize that, we have models from cell lines to transgenic models. So easiest is the model, then less, how to say, information it is giving. Cell lines give you minimal information about disease, and transgenic rats are giving the best information about disease. And what we are doing, we are usually taking the simplest model that gives information. We will test it, whether our hypothesis is correct there, and then we will go to next level model. We are doing, in our lab, we are doing most of the work with neuronal cell lines and primary neurons. So it depends. If we do the imaging, we are doing imaging mostly in axons. Then we don't have axons in cell lines. We can't start from the cell lines. In neuronal cell lines, we don't have a proper axons synapses. So the first level we can start is the primary neuron level. So we will start with primary neurons. If we have something fantastic, then we will go to patient-derived ipsis and neurons. So we have done that with Wolff's butterfly deficiency syndrome. Or then we go to the zebrafish or transgenic rodent models. Zebrafish we can do ourselves, but we have good, also expensive core facility for transgenic models. And it's also relevant that, so if you have transgenic rat model, it's nice, but most transgenic mice models, it's nice, but you should have very good equipment to test the symptoms. So it's if you have transgenic rat but no equipment to see the motor symptoms and so on, then it's not helping you, this model. Only pathology is not enough. So any questions about this general part? What do you think about models? So what I heard that in university you have Animal House, but you don't have transgenic models in Animal House. So this is limitation, because you can't go to this, how to say, last level. If you use the chemically induced models, then it's substitute, but not perfect. Somebody is using zebrafish, Drosophila. Zebrafish is more complicated. You need water tanks and so on, zebrafish facility. But Drosophila is very easy and cheap. There is Drosophila, how to say, banks available where you can buy some stocks having something knocked in or out. But then what you will do with Drosophila, you can't do very much. You can do imaging. People are imaging something in Drosophila. We have not done that. And you can do sometimes very simple behavior also, how they are flying and how they react to some kind of signal. So it's very limited what you can do, but you can do. There are Drosophila models for some neurodegenerative diseases. And so some models exist. Sometimes you can use them. Okay, no questions. Then let's go on. So in vivo rodent models, it's complicated. So you really if you have to decide what kind of model you use tomorrow, then it is a lot of search to find the proper one. So let's start with Alzheimer. So one thing or one model that was used is directly inject synthetic beta amyloid into the brain, hippocampus or cortex. So this leads to rapid formation of aggregates, and you will have some Alzheimer-like changes. So why it's good, you will get it within few weeks. So you can exactly standardize your experiment, how much, where you will inject it. So if it's something which is affecting, if your model, if your experiment requires, is simple and very effective, then this model may work. So it's good for the screening if you have some strong agents to test. But why it's not really perfect? Perfect is that it's so fast. It's never in real life, the Alzheimer is never progressing so fast. And then you don't get really the Alzheimer pathology. You will get acute inflammation and damage, but you really don't get Alzheimer plaques or tau tangles. Your neurons are not dying the same way they are dying in Alzheimer. And Alzheimer is progressive disease. If you inject something, it is just a big insult, inflammation. It's not progressing further like Alzheimer is doing. So it's maybe good for the start again, but if you want to say that your treatment is really against Alzheimer, then it's not acceptable. So you need transgenic model to that. There are a lot of transgenic models available. So the first were amyloid precursor protein mutations. Then came amyloid precursor protein plus presenilin mutation-based mice. And then after that, tau mutation-based mice. So, and each group has a lot of, as you can see, a lot of different options. So what is the best and what is for you? So nowadays they are thinking that the best for amyloid pathology is so-called 5-FID model or APP-PS1 model. So this 5-FID overexpresses human APP and PSEN genes, and they have 5 different mutations, 3 in APP and 2 in PSEN1. So why this model is good? Human genes and a lot of mutations in most relevant genes. APP is this precursor for beta amyloid, and PSEN, the presenilin 1, is this protease cleaving it. So if you have 5 mutations, you will get your disease very fast, your symptoms very fast, and they are most close to human symptoms. Then if you for some reason you don't want to study amyloid pathology, then you want to study tau pathology, then you have these 2 models where you express, but the mice are expressing specific tau mutations which lead to frontotemporal dementia. So there is no amyloid pathology, so you don't have really full Alzheimer's disease. this model is good only if you are studying tau specifically. And then physiologically most relevant is this model called APP-NLGF knock-in, which means that it has 3 human mutations in APP, Swedish Arctic Iberian they call it. So this NL and G and F are somehow related to that. So why it's good? It's not so rapid than the first, this 5-FID model. It's slower, but it's maybe more human-like. So in Alzheimer's is progressing slowly, and also in this case the disease in mice is progressing slowly. So you have to choose accordingly to that. Why the rapid models are better? Rapid models are better because you will get some the results out earlier, and it's significantly cheaper. But then it's less relevant to human disease, so you lose a little bit of relevance with that. So then Parkinson disease models. So there is good site explaining these models in simple way very well. This Maze Contact Science.com rodent models of Parkinson disease general overview. So in case of Parkinson you can similarly have pharmacological models and transgenic models. So what people are using? They're using reserpine, which is having very limited human relevance. Then they use 6-HDA, which is also having relatively low human relevance. And then they use MPTP, paraquat, and rotenone. All these things, they are affecting the mitochondrial respiratory chain, and they induce a little bit similar kind of symptoms. MPTP is good, it is more specifically killing the dopaminergic neurons, because it enters to dopaminergic neurons through dopamine transporter. Paraquat and specifically rotenone are affecting all cells. So there is death of dopamine. First there is inhibition of mitochondrial respiratory chain with these 3 compounds. Mitochondria will produce ROS, don't produce ATP. These dopaminergic neurons are most sensitive to that, they will die away, and there is a loss of dopamine and symptoms coming from that. Again, they are very fast. You inject rotenone, you will get, or administer it somehow else, you will get results within weeks. So it's fast, simple, but it's not really reflecting the progression of Parkinson disease. You have also some alpha-synuclein accumulation. And trichloroethylene, it's not really good because you may have some alpha-synuclein accumulation, but you don't significantly lose the dopamine. So genetic models. Genetic models are alpha-synuclein models. We'll go to next slides. Alpha-synuclein models, Parkin, Pink, DG1 and LRRK2 models. So alpha-synuclein models are best. You have alpha-synuclein accumulation, but this alpha-synuclein accumulates or deposits, they are not looking fully as you have in humans. They are not really have the Lewy body structure. And another thing is that in these models the substantia nigra neurons are not so much dying. So also problems with Parkin and Pink that I will explain later. Yeah, and then LRRK knockout has no neurodegeneration. But if you use the LRRK mutant overexpression, then you will get something. So just to summarize, it was just a quick overview that you don't have really good Parkinson's disease genetic model as well. And then also very quickly about Huntington models, there are pharmacological models, quinolinic acid models, phenitropionic acid models, and then transgenic models. Transgenic models are okay. Then there is always question about the human symptoms and the rat or mice symptoms. So very rarely you will see all human symptoms also in rodents. So there is no perfect disease model in case of neurodegenerative diseases. The symptoms are also different because how you measure exactly anxiety in mice. Okay, motor symptoms are maybe more similar to measure and also more easy to measure using different kind of things. But these neuropsychiatric symptoms are not so easy to measure. For example, Huntington patients have difficulties with speaking. You can't measure that in mice. Then there is also question, mice transgenic models or rat transgenic models. Mice transgenic models are of course most popular and they're much cheaper to maintain than rat transgenic models. But sometimes you have a case, what we have later, where the mice models do not show symptoms, but rat models show symptoms. And in that case, you have no other options than to use rats. Then the animal facilities. So why they are in our countries, why they are so expensive. There are two reasons for that. Of course, everything is more expensive, but one reason is that these ethical requirements are very high, which means that the keeping of the animals, the maintenance, etc., is very controlled, should have very high quality, which means a lot of money. And then another thing is that if you want to publish it, well, it requires very constant environment in animal house, and everything should be standardized. If you do something for the pharma companies, then even it's more complicated. So therefore in our animal house, everything is, not everything, but a lot of things are automatized, as you can see here. The rooms are fully controlled, humidity, whatever is under control. The people working there should have good education, special education, and altogether this is the reason why this in vivo experiment I told you about will cost almost 100,000 euros. But if it is successful, then everybody will accept it, because everything is well documented, well done. Then another thing what is problematic in Western countries that we have these people who want to protect the animals. So in Estonia this is not a problem, but if you take the UK or US, then these people are more serious and they don't like the animal experiments. They don't like the rodent experiments as well. And first is that sometimes some more aggressive groups are a danger. For example, when I had my postdoc, I was visiting students in state. Then the animal house was next to the hospital helicopter pad, and the hospital was very afraid that when these people will come again to protest against animal use, they will also do something bad to hospital helicopters. We don't have such kind of problem, but still people are pressing politicians to make the rules and legislation more strict, to use less animals, and to complicate the use of animals, to make the ethic regulations stricter and stricter. So if we apply for permit, it could take 4 months to get it. In UK, it could take a year to get it. So, and meanwhile, you can't advance your science. So, but this is unavoidable, because if we don't have a rodent experiments, then we will be rodents ourselves. In that case, the drug testing will be done more in humans, or the less tested drugs will reach to humans. Questions about the animal models of neurodegenerative diseases before we will go to example. So, have you used animal models in your research? If you have, please tell what kind of animal models you have used. Please. Yes. And you are injecting beta molecule. Okay. And you said you are using?
Tested at male. I am injecting a chemically induced model of diabetes.
Okay. And other people, you are not doing animal, or?
Sir, I have induced type 2 diabetes in Spandori rats and kept it for 14 weeks to check the cognitive deficit.
Okay, thank you.
So we have yeast model for Huntington's disease, and also we have this mice model, which we use 3 and 3 model. Okay.
So you have experience, some of you have experience, quite good experience. And when you have experience with publishing also the work you are doing in these models, is it the, problem? Nobody is complaining reviewers. So, it's good. So, when I started, and I did a lot of animal work, and I think when I started it was mostly experimental cardiology, and then I moved to neuroscience. Then it was no transgenic models. It was very easy to publish because this was the best what was available. And when the when the transgenic models came, then we tried this Rotenon experiment, I remember. Then already the boss said to me, then no, don't do it. Please just take somehow the transgenic mice in, because this Rotenon is not so good. And then after that, it was maybe 15 years ago, we moved in lab to transgenic animals. But for us it was easy because we have very good core facility and very good behavioral equipment and everything. So if you will somehow have access to these transgenic core facilities, then it will be good to use these. But okay, so more specifically what we are doing. So how to model Parkin and Pink deficiency? So if we want specifically not to model Parkinson's disease, but we want to model early onset Parkinson's disease or juvenile Parkinson's disease, then most of the cases of this, more than half of these familiar cases related to this juvenile Parkinson's disease, starting before 40s, they are related to Parkin mutations. And then 50% are related to pink mutations. And as you remember from earlier, the pink and Parkin were together. Pink accumulates to outer membrane, induces Parkin translocation to mitochondria, and this is initiating autophagy. So what could be studied? What is the easiest functional assay to study that? So they also, if you are looking for the easiest function, or you're looking for the functional assay, you have two rules or two things to consider. First, this functional assay should be simple. It should be reliable. And another, it should be as close as possible to initial defect. So if the initial defect is in pink accumulation, pink is, how to say, not working, and or initial defect is in Parkin, Parkin is not ubiquitinating something, then you should test specifically that if it's easy. So for example, if pink is accumulating or not accumulating to mitochondria, so you can see here the Western blot, normal pink is not accumulating, and then if you depolarize mitochondria, last column, you have a strong band of pink 1 here. So pink 1 is accumulating, Western blot, but Western blot is really not good for the testing. If you have a lot of compounds or you want to do routine job, then it's not very much perfect. Then another thing what is happening next to this pink accumulation is that Parkin is going to mitochondria. Cytosolic Parkin translocates to mitochondria. This is much easier to test. You overexpress the yellow fluorescent protein fused Parkin, and in normal cases it is just in cytosol everywhere. You can see the PC6 cell, if I'm correct here. So it's everywhere. And if you treat your cells with something which depolarizes mito or almost depolarizes mito, you see that this cytosolic is going to some kind of dots, and the dots are colocalizing with mitochondria. And we did high resolution image to show that this is the case. Red are the mitochondria, and this red is surrounded by this green, yellow, greenish Parkin. Just to be sure that these low magnification aggregates are really mitochondria surrounded by Parkin. And this is very easy to do. You are just transfecting your cells with yellow fluorescent Parkin and something else if necessary, and you will have Parkin in cytosol or Parkin in mito. You will count the cells and you will get the numbers. So the experiment is fast. You can study a huge number of different groups and you will get out numbers. In Western blots you have to do a lot of Westerns to get numbers out. Here you will get numbers quickly out. So just an example, in wild type control everything was in cytosol. If we treat with a toxin, alpha is going to the mitochondria. If we have a Parkin disease mutations, then Parkin is not going to mitochondria very well. These two are disease mutations. So it's a very easy functional assay, and it's closest to initial problem. So this is what we did in the simplest system where it could be done. It's cell line. So we got it, we were happy. Then the next step is to show is the same happening in neurons. But we can't do this experiment in neurons so easily because we have toxicity issues. So we were not able to do it in neurons. If we treat neurons with these toxins, they will immediately die away. They are not having enough energy. So in neurons we went to different level. We went to next level. Okay, barking goes to mitochondria. What it is doing? It is inducing mitophagy. So we measured in neurons the mitophagy. Mitophagy means we overexpress the autophagosome marker, mitochondrial marker, and checked whether they were colocalizing. So there again, red are mitochondria and green is autophagosome marker. And we got numbers, we quantified how many we have. Treatment with toxin increased the number of mitochondria inside of lysosomes. So this work was showing that also in more complicated neuronal system we have, how to say, effects. There are many other options, more specific, what people are using to measure or to do these functional experiments in cell lines and primary neurons, but we will skip these at the moment. And then, if you want to know whether the same happens in Ips-derived neurons, then you can do more things. So if you take the cell lines, no axons, then you can measure basics. Do you have a Parkin translocation? Do you have a mitophagy? Do you have a mitochondrial function? But you really can't measure whether you have changes in cell growth, cell survival, axonal growth, because you have different cells, you don't have axons. So you should go to neurons or Ips-derived neurons. Then you can really measure, if you don't have a mitophagy, will your neurons die? Are your axons growing? Are your axons branching? So if it's Ips-derived neurons, then you can do that even with dopaminergic neurons. Are your dopaminergic neurons surviving? Are their axons going? What happens with specific dopamine pathway there? What happens with synaptic function? So you have more options. You are more specific. So we have not worked with Ips-derived PINK1 or Parkin mutations, but it has been shown that they are behaving similarly as expected. But the problem is that they are variable and expensive. I'm trying to explain what this means. So the results are nice. They were not our results, but from literature they were as expected. But what makes it complicated? First is that to grow these cells is expensive. You need special laminin to cover your cover glasses or plastic. You need very expensive media for these stem cells. And since you are growing them a long time, you will lose quite a lot. You will use quite a lot of these compounds or medias. And another thing is variability. So when you are inducing this differentiation, which takes 1 month, then they are differentiating every time slightly differently. There is variability. And then the problem is, what I told about earlier, is that you have to always compare patient and the control. So problem is that your patient and control, one has mutation and another has no mutation, but their genetic background is different. Other genes are different. So you really compare just one control and one patient because of difference. So you have two options after that. You can have a minimum 3 control patients and the 3 patients, all with different backgrounds, and all, if you get statistics after that, then it's okay. So your N is patient or human. So this is quite complicated. You need 6 cultures running together. Or a simpler, more expensive maybe option is that you will take this patient mutation and you will correct it by CRISP. In that case, you have 2 groups. One is the patient, or 3 groups, you still have a control, but you have a patient and you have patient with corrected mutation. Then the difference only is this mutation. Genetic background is the same. Then it's easier to do. You need 3 groups, 2 groups, but this additional step is maybe, if company is doing it, it's 20,000-30,000 euros again. So this is all very important, but it's too expensive. So for us, we don't have enough money to do these things properly. It's for the big labs in US and UK and Europe. Then if we have a pink and Parkin, we have a zebrafish model, we have Drosophila models, we have C. elegans models. And since it is motor deficit, we can study it more easily in Drosophila. So the flies have some kind of flying defects, muscle degeneration, and it's visible how they are flying. So if you have Alzheimer, then it's maybe more complicated to have Drosophila model. But okay, people don't use nowadays very much for these models because they are too far from humans anyway. And then explanation, what is the problem with mammalian models of pink and Parkin deficiency? We had pink deficient and Parkin deficient, or actually it was pink mutation, mice in our animal house 10 years ago. And we were hoping that we can study whether the one target we studied at that moment is giving some protection. What was the problem is that we had no phenotype. We had to wait 12 months to see very small changes in motor behavior, and they were really very subtle. So this means it's not doable. If you want to test it, you have to wait 12 months, and then your changes are very minute. What you can do? Nothing. So we stopped that just experiment, and the money was lost. So let's start with Parkin. So if you take the Parkin knockout mice, then you see there is no really phenotype. Dopaminergic neurons still there, alpha-synuclein not aggregates, locomotor dysfunction, no dysfunction. But then if you have a complicated model, you have a mutation, Parkin mutation and rat, then you have all these 3 phenotypes. Dopaminergic neurons die, alpha-synuclein aggregates, locomotor dysfunction is there. It's also not fully correct because alpha-synuclein aggregates, but if you take the patients, then it's not so frequently aggregating at all. Or if you take pink deficient mice, again you see no effect, no effect, no effect. Then you will get some effect with rats. So in case of rats, there is locomotor dysfunction and dopaminergic neuron change. But it's also not fully perfect. But if you go then to monkeys, you see macaca mulata, all these things are positive. So this means that you have really to check whether the model you are planning to use, transgenic model, has sufficient phenotype, and also that more complicated is the organism, then is more likely to have a phenotype. Mice are not so sensitive in this case to this pink and Parkin deficiency or mutations. Rats are, so you can work with rats. Rats are very available from Jackson lab. Okay, you can't work with monkeys. We can't work with monkeys. And what this means that you are working with these animals. So this means that you are doing different kind of tests. In case of Parkinson, it's the motor tests. So that you are, okay, what we did with this pink and Parkin deficient mice. We use simple open field tests. This is just a box. Mice or rats are moving here, and you have a camera, you will analyze the movement pattern, you will see how much they are moving, what is the speed of moving. Then what is very good is the rotarod. This is just circling. You will put the rat or mice in, it will start to circle, and you are just checking when it is falling down, how well, what is the motor coordination and balance. And then most sensitive is the gait analysis. This is just this, how to say, running belt. Mice, rats are running or walking there, and the system measures how they are doing that. What is the length of the stride and the gait. So if you remember, Parkinson patients had that problem, so it's very close to measure this gait and walking and running of the mice and rat patients. So most sensitive and very quantitative. And then the grip strength is just to measure how well they could grip to something. This is less sensitive, but still good. One test is never enough, so usually you will do a lot of tests. So we did all these 4 tests when we did our experiments. So it was good. Just the final figure here is that there was one paper and a few more papers saying that pink one has nothing very much to do with the mitophagy. But anyway, most of the people think that it's relevant for the mitophagy, and mitophagy is the reason for Parkinson's disease in this specific case. So this is about the models. Do you have any questions? So can we have a 5 minutes break then? Okay, just 5 minutes. Then we will have a drug development. Morning.
We can have a tea break right now.
We can have a longer also. It's up to you or up to them how long they because then the next lecture is maybe slightly shorter, 40 minutes or so. We can have a break after that, or now. What is better? Because if you have tea already here.
We don't have
okay, then.
So I can tell them to delay it.
Yeah, then it's. Or we can make a break also inside of lecture, so everything is okay.
Yes, it is.
Just tell me when it's okay to.
It's all on you, so I
Okay.
can stop them from tea right now.
Okay, then maybe then
Or it's okay.
after 40 minutes or. Okay, we can continue then. So it was good to have discussions with you, and 2 additional points to previous lecture. So that if you work in, at least in Europe or UK, with animals, then you need a license to work with animals. So in different countries it's different. You always need a license, but the question is how strictly or strongly they force it. So in Estonia you need a license. To get the license you should pass the course, local course, 2 weeks. They are teaching you everything, how to touch the animals, how to kill the animals. And after that you have official paper, you can work it. Or PhD students working with animals, they are passing this course. Sometimes it's a less strict, so that if you do some animal experiments but your supervisor has a license, then you are considered as a helper only and you can do it. In Estonia it works, in UK maybe not. So and the question is whether they accept licenses from outside. In Estonia they don't want to accept. If it's from EU then okay, but if it's from the third country then they don't care very much. I don't know about Germany and other countries, so but if you have already the local license, it's anyway easier to pass the course in the country you are going to PhD or postdoc. Then another thing is that I told you that you usually start from simple model and then you will go to more complicated model. So this is what I am doing. But I know very good people doing it very differently. We have one cooperator, very high quality researcher from Taiwan. They have very good transgenic core facility and they are very experienced to make the transgens, to study them. They are doing it differently. If they have some compound which could be good, they have some target which is very relevant, interesting, they will first check it in vivo in mice. Is it really working there? And then they will go to study the mechanism. And it makes also point, because when I'm doing my research 5 years, I'm studying the mechanism and then I discover that there is nothing in vivo, then why I did it? So she's quickly checking, is it working in vivo? If it works, then she will spend 5 years to do in vitro experiments, and she can be sure that this is a really important thing what she's studying. So that vice versa is also possible, not just as I told before. Okay, so let's go to drug development. So it's now very generic what I'm speaking, but I have a feeling that most of the researchers need that general knowledge as well. So it's about drug development pipeline, then that you always should know what will be at the end. Then what you do, you should understand what is the unmet clinical need if you develop something. Then it's about target product profile and a little bit about the current developments. And then we have this seminar after that shortly. I will give you for groups some rare diseases, and you will have to develop or do this target product profile for these diseases. So the drug development pipeline is this classical tube that you have discovery phase, then preclinical phase, then in states you have this investigational new drug application to FDA, then you have the safety, clinical phase safety and clinical phase efficiency, and then after that you may get drug registration. So the first phase where we are working, this is discovery and development, we will need targets. We need targets which we can modify, which will affect our disease. Then we need compounds to that target. We will do the testing, screening, whatever. Or it could be an opposite, vice versa as well, that we have some kind of drug candidate which is doing something interesting, but we don't know how it works. This is not enough. We should have its target, or we should at least know what it is doing, what is the molecular mechanism, what is the biological plausibility, if it's correct phrase, for that drug effect. If you have a just compound which is not working, which is working but you don't know what it is doing, then it's limitation already. For example, this is limitation with these plant-derived compounds, that it's very difficult to understand what exactly they are doing in this cell. You don't get this mechanism so easily, and this limits their relevance a little bit. If you have a compound, you have a mechanism, then you should be sure that is it possible to administer it? Is it absorbing? What are the interactions? And you can develop that further to make it better. Then, and what could be the possible side effects? Then you will have to do the testing. You should have some kind of tests, in vivo tests in animal, to show that this compound is protective. Then you should do the proper toxicology tests to show that it's not too dangerous, it's safe. And all these toxicology tests at the end, you should do following the good laboratory practice GLP, which is quite complicated. Usually this phase will be outsourced to specific company doing toxicology. And then in States at least you will submit this investigational new drug application, and you can do the clinical research after that. At the beginning phase 1 it's safety, small amount of people. Then phase 2 it's the efficacy, how it works in small amount of people, and phase 3 is a bigger test. You will see efficacy, safety also from there. So after this third phase you are able to register it. So that it's slightly more complicated. The phase 2 could be 2A and 2B, but okay. So, and if you have at the beginning 10,000 compounds, then in preclinical phase you have 250 compounds. Then in clinical phase you have 5 compounds. Then you are lucky if at the end you have 1 compound that will reach to approval. So there are 2 agencies we consider most important. One is the US Food and Drug Administration, and another is European Medicines Agency. So why they are for us most important? One thing is that the biggest markets are there. So if you have a new drug and you really hope that it is doing something and it is making money, then you should start from the biggest markets. You can do it also locally here, I think, because you have a big market, but in Estonia you don't have market, 1.3 million. For us it's Europe and US for start. So they are checking whether your data is good enough. So if you will submit all your information to these agencies, they will check, is it safe, is it effective? Is the risk benefit positive? And is it making sense to have one more drug in the market? After that, you can go to manufacture, marketing, and selling. If something happens after that, the side effects, unexpected side effects are coming, then it will be called back from the market. This is not happening so frequently, but it is happening sometimes. So this is the pipeline. So usually what happens is that academy is at the beginning doing basic research. Then in discovery phase you already have academia and industry. And from preclinical, clinical phases you have mostly industry. So industry is usually interested only when they are sure that it is safe. They will take it over maybe from this place here. And then basic researchers, they usually don't want to do too much. They don't want to go to preclinical toxicology. They don't have money for that. They can just do the basic research, show the efficiency, publish it. So this from this from drug discovery to clinical development. So this part is called frequently the valley of death or translational gap, because nobody wants to do it. Academy, no money; industry, too big risk. So it's not how to say always like that. Big pharma companies, also small pharma companies, are doing quite a lot of basic research, or maybe not basic research. They are at least doing a lot of drug discovery. So it's of course just oncology drugs. They are summarized. It's not good example, but it shows that pharma companies are even in discovery phase. Pharma companies are more involved than academy, and then at the end it is only long. So that academy is usually finishing here or here. Not because they don't want to have a drug in the market, but just because they have no resources for that. So, but if you are starting from here, you are trying to find out that you have some drug, then you should understand exactly anyway what happens at the end. Because why to develop some molecule to spend a lot of time for optimization and so on, if you don't care what happens with it at the end. So that usually you publish and nothing more, but if you develop it further, then it makes sense to develop it further only if it has potential to be a drug. So you should understand that potential. So there are a lot of things you should know. You should very clearly know what is the unmet medical need. You should know what is the expected outcome, how efficient it is. You should know what is the possible side effects, what is the risk of side effects, and are these acceptable. Then what people are not thinking, it's delivery and dosing. You may have a very good peptide in cell culture, but how you will deliver this cell penetrating peptide if you have a patient? And then money is also relevant. If you have a very expensive therapy, then who is paying? if you have cheap therapy, and even if the expensive one is in the market, your cheap one is competitive. So what is the competition? And what is the total cost? So if you are developing something, you should understand that somebody later will have to invest to this project. You can sell it to university, your university will sell it, or whatever, but you should understand that there will be buyers somewhere. And it's how big is the market, expected market. If you have a Wolfram syndrome, we have it, and then it's small. If it's Alzheimer, it's big. But then it's regulatory path. If you have a disease which have already the drugs, then this regulatory path is long. If you have rare disease, orphan indication, rare disease, you will get your drug to market much faster. Or if there is no treatment, you can get expedited review, as we had in this case of fileless drug in last lectures. Okay, then you should understand exactly also what is the IP status, so that patents are key components in drug development. If you have a drug candidate, then you should patent that, or your university should patent that at some moment, because the patent says that this is your drugs and there is something to sell further. So if you don't have a possibility for IP protection, for whatever reason, it's not novel enough, your drug, or you just don't care about patenting, then it's and you publish it before, then it's absolutely clear that this drug will never reach to the clinic. So that if you publish before, or you will even speak about it in conference, or I will speak about it here and it goes to the internet, then I can't publish, after that I can't patent this anymore, because it is known. What is possible is, which is slightly tricky, but you can patent and publish together. And what is less tricky and absolutely safe is that you will publish year and a half after patenting. If you have to speak about your drug and its nature before you patent, then it's necessary to use confidentiality disclosure agreements. This is removing the risk that this goes to public. Not fully, but more or less. So what you need, your drug candidate, what it needs to be patentable. So you can patent it if it's clearly new compound, or at least it's novel compound. There is slight difference here. So something is absolutely new and something is a little bit less new. So then if it's not absolutely clear that this is, it looks like invention, it's not obvious. This is not concerning drug candidates so much. Then it should be useful also that you can patent something which could be useful to somebody at the end. Then there are something you can patent. You can't patent gene sequence or protein sequence which is in the cells. And then most important is that to patent something you need very good data to support that. You can't tell that it's, this drug is good against Alzheimer, but you should describe it in very great detail to have convincing data, like in publications, to show that this is really protective. You should explain how this drug works, how you get this drug, and so on. So it's not so clear when and where to patent. If you patent too early, you don't have enough data, then you may not get the patent, or you don't cover all aspects, and later somebody will develop it further and do the new patent. If you patent too late, then it's risk that there are so many researchers in the world that somebody is doing the same thing, and you are not able to patent anymore. Then it's a very tricky question where to patent. So, if you will have a new drug in Estonia, then you can patent, or in India, it's the same. Then you can patent in your home country. Then you can patent in some other countries, US, UK, Japan. And then you have such kind of additional places. European patent, unitary patent, and international patent. I will explain them because it's an important issue. And timeline of patenting. So priority filing is the time you submit your patent application to your first patent office, wherever it is. Then time starts to go, and you will get this 12-month period after during what your discovery is, how to say, safe. And you can, during that period, to submit it to other patent agencies, patent offices. And this priority filing date is showing its legal timestamp, saying that from this date as of this date I invented this. So after that, theoretically, you can publish it everywhere, papers, conferences, web, with small risks. So, but this is only one country. If you do it only in India, then it's not covered in US or in Europe. How to cover it in every place? It's not so easy. In this figure, it's this PCT filing, or it could be one US or EU patent. So PCT filing is that this is the Patent Cooperation Treaty, international treaty covering most of the countries. You can see the blue here. So you will give application, you will file it there, and you can do it only within 12 months after your first patent application. So people may think that after that I will get a patent in all countries, but this is actually a mistake. You don't get anything. You will just get extension. You will get maybe 30 months to patent it in all other countries you want. So it costs money, it doesn't grant you any rights, but it gives you time, because otherwise you have only these 12 months to submit it to other places. But if you have this PCT application also, then you have altogether 30 months. In some countries slightly less, in some countries, in India I think it's 31 also. So you will buy time for your patent applications. You don't get the patent. Then what you really want, you want to patent in big markets. US, Europe, UK, Japan are actually preferable for us, in Estonia at least. So we can have a European patent, or in Europe, which covers all Europe, or we can have a unitary patent, which covers half of the Europe. but which is much easier to get. We have not to translate it to 20, 30 different language. We don't have to pay a lot of money to deal with local people. Unitary patent gives us this half protection. You will see the red, orange place here, but fast and cheaply. So, what it is costing? If we have only US patent application, then it's maybe 10,000 euros. If we will have a local and after that US patent, then it's maybe up to 15,000 euros. If we will have Estonia, this PCT and US, then we have it up to 25,000 euros. Or if we have just main markets, then it's 35,000 euros. So it's quite expensive. And if you are developing a drug further than is your direct academic interest, then you should think about that, who is paying this. So but after that is also the patent managing costs will come in. But first is the filing costs. That if you really want to protect it in all main markets, you need a lot of money, and usually university is not giving you this money. If you establish some kind of small company and university give rights to this company, then this company should have this money. So we don't have this money. Once we had this situation, we had something to patent. We made application only in US because this is the biggest market. If you have it protected there, then and it's somebody will be interested, then you have some time to protect it somewhere else as well. So it seems to be some kind of legal story, but you actually, if you will do something, you will make application, grant application, then you should know everything that. Because at least in our grant applications, in EU grant applications, even in Marie Curie fellowship grant applications, you have the section IP management, and you can't write to IP management, yeah, if I will have something, I will patent it, or our university will patent it, because this means nothing. Usually these applications expect more information, and to write more information to this application, you should understand the patenting logic, timelines and costs. If you will get these applications submitted, then there will be examination of the patent, which means that it could take first round half a year, but several rounds could be several years. So you will have a reviewers actually quite similarly to journals, except that usually you have one patent reviewer in US. Quite similarly to journals, they are checking, is your finding novel? They will check the literature. You have claims in patents. You claim this is doing that. They will check, has somebody shown that before? And if they will find that it was published before, then it's not novel. You don't have anything. So in many cases, the examination says that it's not novel and you don't get it. Even if you pay the money to the patent attorneys and the patent offices already, but you don't get your patent. You have just submitted application, which you can use in CV. It's good for CV anyway, if you have a patent application, but it's really has no commercial value. If you get this patent or company or university, then it could be 20 years of protection, or it could be 25 years if you could extend it. your discovery is protected for 20 years. But it's also not really fully protected, because other people can invent something how to cross your patent. If you have a compound patented, they will add few groups and they will get new patent. So then you have nothing, but you will sell if there is another patent. Maybe the compound is even better what they have. If the pharma company wants to develop similar drug, they will modify your drug a little bit, and it's their drug after that. So that in patent application you should try to cover your discovery so well that it is difficult for others to cross or pass your description. Okay. So then another thing, you should very well understand the clinical need. So you should know, do you have patients who need your drug? So if we are speaking, for example, Parkinson's disease, then you can have very different kind of drugs. The best ever drug is the drug which stops the progression of Parkinson's disease. Or it's also good to have this levodopa, which is blocking the symptoms of Parkinson's, motor symptoms of Parkinson's disease. But then you have also other problems. Parkinson patients have other problems. One of the non-motor symptoms is the constipation. 70 to 80% of Parkinson patients have it. And it comes sometimes before the motor symptoms. And there is drug candidate currently in clinical phase, if I'm not mistaken, which is theoretically at least effective against this constipation in Parkinson patients. So that the drug could be also against of these minor symptoms, which is problem. And if 70 to 80% of patients, Parkinson patients, have it, then it's a huge market, and constipation is a real problem. People will start to take this drug if it's effective. So that you should have, if somebody asks you what is the clinical need against Parkinson's disease, in case of Parkinson's disease, it is not just this what levodopa is doing, and so it's simple things. It could be also such kind of very different minor thing, which is problem for patients. So you should understand it, what patients need. Then another thing which is clear to understand is that what is the disease burden. That it's clear that Alzheimer's disease, having 50 million patients worldwide, is a very big market. So if you take the US Medicare Medicaid costs, so it's here it's maybe in billions, 200 billions per year. It's a really clearly huge market, and it's everywhere where there is money, there is need, because it's disease is everywhere. But then if you take a rare disease, what we're studying, Wolfram syndrome, it's more or less 1 patient per million worldwide. If we are estimating how many patients we have, it's 10,000 patients at the moment worldwide. Disease is very nasty. It starts with diabetes, then it's the optical atrophy, hearing loss, neurological symptoms, and so the time people are having very low life quality is 10 to 20 or more years. To treat these patients is very expensive. It's difficult to to say how expensive it is, but what I found from one orphanet journal paper, that in UK the lifetime cost of treatment of this patient, one patient with Wolfram syndrome, is 1.5 million USD. So of course worldwide the treatment cost is not 1.5 million per patient, but if it would be, then it's 15 billions already. So that even the rare diseases have a market. So you can say that there is no sense to develop drugs here. There is market if disease treatment costs are at the moment already so high. Then side effects and efficiency. So Alzheimer drugs, at the beginning symptomatic drugs we covered, and then now we have the antibodies, which are disease modifying therapies. What is the difference between the symptomatic drugs and disease modifying therapies? So in case of Alzheimer, you can see the graph here. This is the cognitive performance. Normal aging, it goes slightly down. If it's untreated Alzheimer, then it declines, going to close to zero with time. The aim of disease modifying therapy, if you get the diagnosis at that point here, then the aim of disease modifying therapy is to stop progression. This is perfect. The symptomatic therapy, this means you will get some improvement, but progression goes on. You have some improvement only. So it's a very clear difference. Symptomatic therapy, disease modifying therapy. Disease modifying therapy is always preferred. Then another thing is efficiency. So this is the Alzheimer, Leqembi Alzheimer antibody. So we have here, can't read it, but some kind of parameter. 0 means everything is okay, and here everything is not so okay. And you see that without antibody there is disease progression. Things are going worse. With antibody, things are not going so quickly worse. It's 27% difference. It's not too much. It's okay, but it's not too much. If you will have another antibody coming to the market, you are working with another antibody. If your new antibody will have also 27% protection or 10% protection, then nobody is interested. If it's less effective, why to use it? Well, there could be other reasons, but if it's as effective, then why to shift to another antibody, only if it's cheaper? But if your antibody has a 50% protection, efficiency much better, then it's perfect. People will forget this Leqembi. They will start to use your antibody. Then you have side effects to consider. Actually, this is very important. Big part of the efficient therapies don't go from preclinical to clinical phase because you have side effects. If you take the same Leqembi, then what is the problem? This Leqembi, as well as other antibodies, they are leading to amyloid-related imaging abnormalities. It is ARIA with edema, ARIA-E, and ARIA with hemosiderin deposition, ARIA-H. As I told, usually not a problem, but sometimes life-threatening, serious intracerebral hemorrhage, for example. And it's visible in MRI. Okay, you can see in MRI, this is a figure, it's not MRI image, just a figure illustration, but it's visible as it tells, it's imaging abnormality. So let's see, will we get the video also?
Anti-amyloid antibodies used for the treatment of
No, we don't get the video.
often associated with amyloid-related image or ARIA, such as edema and hemorrhage. While the exact mechanism behind this
So it's not
pathogenesis
good day
remains
today, so we will not
ARIA
get
can
the
occur spontaneously in
voice.
Alzheimer's disease and has been associated
But okay, we will
with
manage.
seizures. Given the known
So that this area, so we will see it, areas will have a different symptoms. Headache, visual disturbances, confusion, focal symptoms. And this was the initial problem with that, or maybe you can help with it. Was working yesterday.
201 trial. Analysis of data from the core study. A modest incidence of
So it's coming out from
with
computer,
both
but it's
bi-weekly and monthly
going.
lecanemab dosing regimens, and an even lower incidence of symptomatic
something is
cases
coming.
of less than 3%. Radiographically, this incidence was mild to moderate in severity, with higher occurrence of ARIA during the early stages of treatment. Anti-amyloid antibodies used for the treatment of Alzheimer's disease are often associated with amyloid-related imaging or ARIA, such as edema and hemorrhage.
I can use this microphone here.
ARIA can occur spontaneously in Alzheimer's. Anti-amyloid antibodies used for the treatment of Alzheimer's disease are often associated with amyloid-related imaging abnormalities, or ARIA, such as edema and hemorrhage. While the exact mechanism behind this pathogenesis remains unknown, ARIA can occur spontaneously in Alzheimer's disease and has been associated with cerebral amyloid angiopathy. Incidents of ARIA can often result in symptoms like headache, visual disturbances, confusion, and, infrequently, focal neurological symptoms or seizures. Given the known occurrence of this adverse event with anti-amyloid antibodies, understanding the ARIA profile of emerging new anti-amyloid therapies can help in better therapeutic management. In this study, researchers from the USA report the in-depth ARIA profile for lecanemab, a humanized immunoglobulin G1, IgG1 monoclonal antibody, which has demonstrated efficacy in treating early-stage Alzheimer's disease. The researchers used data for 2 dosing regimens from the core and open-label extension studies of the double-blind, placebo-controlled lecanemab study 201 trial. Analysis of data from the core study revealed a modest incidence of approximately 10% ARIA-E with both the biweekly and monthly lecanemab dosing regimens, and an even lower incidence of symptomatic cases of less than 3%. Radiographically, this incidence was mild to moderate in severity, with higher occurrence of ARIA during the early stages of treatment. Moreover, the exposure-response analysis revealed high incidence of ARIA-E in ApoE4 allele carriers, particularly among the homozygous carriers, and an increase in ARIA incidence with increasing dosage of lecanemab treatment. The analysis also revealed a relatively low incidence of ARIA-H in the core study, which was dose-independent, occurred more commonly in ApoE4 carriers, and without the incidence of any symptomatic cases. Further, consistent findings were observed in ARIA incidence among the OLE participants, with 1 case of symptomatic intracerebral hemorrhage greater than 1 centimeter. In summary, these findings establish the safety profile of biweekly lecanemab dosing regimen with...
Okay. So they made this study, and from this study they concluded that in normal traditional patients the probability prevalence specific side effect is not so big, 6% if I remember it correctly. But then in some subpopulations it is higher, so that these ApoE homozygote patients, they had higher risks. So if you remember, this ApoE was risk gene for Alzheimer. So they have a higher risk for serious ARIA also. And because of that, it is, how to say, said that this ApoE status should be checked before the antibody treatment. And if they have these ApoE mutations, then it should be seriously considered whether the antibody therapy will be used or not, and patients and doctors should discuss the risks. So this is how the information from clinical trials will go to the guidelines. And what this means, if you're developing new antibody, this means that if your antibody is working better than this one, it's good. But it's also, if your antibody will not have this ARIA risk, or even if your antibody doesn't have the ARIA risk in this ApoE homozygotes, then your antibody is already having market somewhere. You can sell it to these specific patients. So antibody sometimes, or another drug, might be even less efficient than previous drug, but if it is having less side effects, then the risk-benefit ratio for this subgroup, for example, is better. Then administration route. So it's easiest way, it's pills and injections. Okay, you have infusions and other things also, but it's usually pills versus injections. So tablets are better, usually, because people don't like injections, they don't like needles, they like pills. But if you use, for example, GLP-1 agonist, you know GLP-1 agonists, what they are doing, diabetes drugs, obesity drugs. So you can have, mostly people inject them once per week, maybe, semaglutide, if I remember it correctly. But you can have also the tablets. So people who don't want to inject, they can use the tablets. But the problem is that this tablet goes to the digestive system, absorption is not perfect, it's slower, and your effects are weaker, or they come later, they are weaker. Sometimes these semaglutide have some kind of strange side effects. Of GLP-1 injections, it is like that, but in general people prefer tablets if possible. Then for all new drugs you need to know the pharmacokinetics. PKPD is the abbreviation, very quickly, what the body is doing with your drug, because if the half-life is 5 minutes, then you don't have a drug, or half-life is 1 minute, because you have to take it 24 times per day. It's not working. Half-life should be long enough. Distribution should be good enough. If it's your Alzheimer drug and it's not going to through the blood-brain barrier, then you don't have a drug usually. and then metabolism and excretion. So if your drug is having very long, it starts to accumulate to somewhere it is maybe not good, it may have bad metabolites and so on. So a lot of different things you should know very quickly. Then there is price. So if we take the same antibody, currently Alzheimer antibody, then its list price year ago in US was 26,000 USD. And then it was calculated in Lancet medical journal that if all European patients will start to use this antibody, then it will cost to EU to 130 billion euros if the price will remain the same. And this is half of the money used for pharmaceuticals in EU. So it's also, this means that there is no hope to treat everybody with this drug in EU, and if somebody will come with drug which is twice or more cheaper, then it will take the market, or at least the price of this Leqembi will go down. So if we have a Parkinson disease, then what is the unmet clinical needs? What do you think? Do we have drugs available? Yes, we have drugs available. Do we have disease-modifying drugs available? No, we don't have. Are existing drugs effective enough? So it is so-so. Levodopa is at the beginning quite effective. Do the existing drugs have unacceptable side effects? Yes, you remember these dyskinesias on-off coming after long levodopa treatments. Are existing drugs hard to use in case of Parkinson disease? No, they are okay. I mean, levodopa is in tablets, no problem. So if you know everything about your disease, listed here, then you can do the target product profile. This is important thing, target product profile for people developing drugs. This is the desired profile of your future drug aimed to treat disease. It has slightly different meanings. In companies, it is the planning tool that you will have to reach to this and this, you have to achieve these and these things, then your drug development is going well. In regulatory agencies, this means that in US to get this permit to go to clinical trials, your drug should be good enough, better than as good or better than previous ones, and this TPP is showing that. And in context of funding agencies or public health, it's also relevant, because if you are having grant applications, you are applying to get money to get for new treatment. You should explain there very clearly why your treatment you are developing will be better than current treatments. So if the disease has already many drugs, and all drugs have some kind of limitation, and your drugs theoretically will not have, then it's a big thing already. If current drugs are perfect, then your drug will not help further. So it's a planning tool for everybody. And it's not just a one-time paper. This is paper you can change during your development process. Just examples. few examples. One example is the pain related to diabetic neuropathy, and it comes from NIH, National Institute of Neurological Disorders and Stroke homepage. So that first thing is that what is your disease, what your drug is doing. You have here the ideal results and minimum acceptable results. The ideal results is that all patients will have no pain or have less pain, and minimum is that here it's the same, but minimum is that what this drug should minimally do to be better than current situation. Then you should have a patient population. So ideally it should treat all the patients in this subgroup. All, for example, diabetes patients. And then what is your minimum? Here it's same, but in real life the minimum could be that it is affecting at least part of the patient. Then what is the treatment? Here it's chronic in both cases, but again, ideal is that it's one treatment just once. So if you take a gene therapy, it's only once. If you have antibody therapy, it's chronic. Delivery, it's again same, but ideal is maybe the pill, less ideal is injection. Then how frequently? Less frequently is preferable. Then what is efficacy? Best ideal result is, of course, that most of the patients will have a relief. But what is the acceptable minimum? 1/3 of patients will have relief. Here it's 30%. And side effects. Ideally almost no side effects, and minimum is that how big they could be. And then what is the therapeutic modality? So this is such kind of official. Then I made myself for Parkinson. So that if we have indication, so the best or ideal is that we will have neuroprotection, disease-modifying therapy for all patients. Minimum is that we have symptomatic treatment. Ideally is that we have all patients, old and young. Minimum is that we have subgroup. Then it's efficacy. We have some kind of endpoint. So it's significant improvement of motor symptoms, maybe 100%. And then primary is just statistically relevant improvement. And safety, safe compared with current dopamine agonist treatments. Treatment route, oral once daily, oral best, not so best, twice daily. And the price, cost effectiveness. Optimum is cost effective every way, minimum is cost effective compared with current therapies. So then pipelines. Just frequently the pipelines for drug developments are how to say, craft like that. Parkinson, Alzheimer. And you see that in the middle we have a phase 3, which is the last clinical phase, then phase 2, and first safety phase, phase 1. And then we have sectors, what they are doing, cognitive symptoms, or neuropsychiatric symptoms, this sector, or disease targeting, disease modifying, small molecular drugs and disease-targeted biologic drugs. And here we have a colors show the mechanism. Affecting amyloid, for example, is red. So that these graphs, they are available for the major diseases. They are very, very good. So you can see from here what is in clinical development, what is in pipelines. You can see how far they have reached, what is the mechanism and what is the general symptomatic or disease modifying, or what kind of symptoms are modified. So they are very good. If you have a rare disease, then you don't find such kind of graphs. Then you are looking for something specific, because these graphs, they are not fresh, they are a little bit old. Then you should go to the clinical trials database. So it's not including everything, but it's the US clinical trial database, which is the best. So I was asking about the clinical studies related to Wolfram syndrome. I got 11 ones. So they are telling what is the drug, they are telling who is doing this study, and what is the situation. And sometimes if the study fails, then it's usually not written here, at least in cases I checked. You can find it from somewhere else what happened. But you will get understanding what is under development. Then there is one rule, fail fast rule in drug development, that you should not go beyond your academic interest if you can't see the unmet need. You can see that your drug is better than others, then just publish it. So if it is not more effective or it has serious side effects, then no hope. Or if you have something which can't be delivered, or if you already can see that there is no hope to find the money for patenting, or you know that university is not giving any money for patenting, then again, why to go on? Just publish and stop it. But then it's a problem that you may have a very good compound, you publish it because you have no money, and your compound will be not developed further by anybody because it's published. This is very sad situation. A lot of candidates will not reach to the clinical trials because there is no money. And this is why, where we had it, this is what is, sorry, this is why this part is called this valley of death. The drugs will die here. So this has been acknowledged little bit by governments. They give the money for this translational phase, and even more from private sector. So there are many private sector, NS, Spark, a lot of such kind of private foundations who specifically, Fox Foundation, others, specifically UK Parkinson, specifically funding this step. And these foundations, they usually not take your discovery, they just give you money for that step. They want that if they consider your discovery relevant, they want that this will somehow reach to clinic. They don't care how exactly, but if their money helps, then they will fund it. But they are very strict. I mean, you should have really good discovery. So we have next coming, I don't know, after lunch, the seminar. And for seminar, I have 11 diseases. I can add few more if necessary. And I would like you to form a groups of, I think, how much people? Groups of 3. You will take one of disease and you will generate for this disease the target product profile according to this table. So this is just example. You will, how to say, remove this part from here what concerns diabetic neuropathy, but you will have to fill this table accordingly to this disease you will get. So now we have a lunchtime, or we will go to we will continue now. What you prefer?
I think they should do this literature survey using
So that so if you will do this table, then you can come and show this table here to others and very quickly, I don't know, up to 3-4 minutes explain what you have in this table. So you need at the beginning maybe some time to prepare, at least 20 minutes, and then
we can meet at 2:30 then.
yes, okay.
Then have some lunch and then we can do
Okay, yes, okay, that's good. So you can take a picture from this. Actually, you should take picture from that, but we should divide you into the groups, and you should pick the disease. Yeah. Otherwise, it will be the same disease.
So all students will pick, I mean, we'll make a group of 3 and each one of you, I mean, each group should pick a different disease. So what we can do is we'll keep, put all the names of the participants in the group and every group will select the disease, and please do it right now in on the WhatsApp itself. Alright? So do the literature survey, whatever you can find, make this table. After lunch, you will be talking about these rare diseases, what are their symptoms, what are the products available for them, and what are the ideal results of those products in those particular diseases that you have selected. Alright? So first you will give whatever you have searched from net, and then Professor Kasi can elaborate that.
You will make the list. Okay. Okay, I will put everything to one slide. So you have disease and the table, and also a few sentences about the this disease cause, symptoms, life expectancy, sentence about current treatment, and then a table. There is no need to be absolutely correct. It's just necessary that you understand what this table means. If you will do something, whatever you will have, you can have a gene therapy or whatever there. You have full freedom, but just make it logical.
It's clear to everyone. So we can meet at 2:20. We'll start the next session by 2:20.
Okay.
We can
Okay.
do some research.
way to connect, or you don't have a hot and okay, you have.
So I'll be discussing about the target product profile of Leigh syndrome. So Leigh syndrome is an inherited neurometabolic disorder in which mostly gene related to oxidative phosphorylation in mitochondria and nuclear DNA are affected mostly. So it affects mostly infants, but later onset is also observed in adults. And after onset of the disease, lifespan is limited for around 2 to 3 years. So this is the TPP which I prepared. So currently there are no permanent treatments available for the disease. Current treatments are mostly symptomatic, which only treat the symptoms of the disease, which include mostly antioxidants and vitamin B1 supplementation. So this is a TPP which I prepared for my hypothetical drug, which I'll be preparing, which I can name as LS Thera. So this is the.
So if I can ask, just
Yes.
what do you mean with efficacy? The survival, improved motor function, ATP levels?
Efficacy.
Yes. I mean that. Survival. If they will survive longer than it is already. Do you mean with that symptomatic treatment, or do you mean with that treatment which is stopping progression?
I'm trying to, sir, actually symptomatic treatments are already available.
Mm-hmm.
So I'm trying to develop a treatment which can address the root cause.
Uh-huh.
So what I'm trying to do is I will have nanoparticles which will have the I can use gene therapy or something loaded on those nanoparticles, and they can address the gene which are affecting the oxidative phosphorylation.
Okay, so you mean with these nanotherapeutics, you mean gene therapy?
Yes.
Okay, so it's a good example because if you go back to previous slide.
Yes, sir.
So you are saying here that you have mutations, many different mutations in nuclear genes, which means that theoretically, if you have such situation, you should have, for each mutation, you should have individual therapy. It's quite complicated. You need a lot of therapies in that case, because they are different.
If we address, then it should work at infant stages, or if we know that the mother is carrying the defective mitochondria, then we can take precautionary.
And then another thing is that you have here mitochondrial genes also. So it's two interesting points. If you plan to have a gene therapy, you're going to have a gene therapy against nuclear mutation. But currently you can't have a really gene therapy for mitochondrial mutations because it's mitochondrial DNA. How you will change that with current tools? And another interesting case here is, if you're speaking about gene therapy, is that what is the current legislation that you will have a gene therapy against one specific mutations, you will do all this development registration, and then if you have another mutation in another gene, you need to do it again against other mutation. So they are planning to make it easier in future. Maybe it's already happening, that you register all these possible therapies once, and if you will have then something against slightly different mutations, you don't need to do paperwork again. So therefore it's a good example disease to explain that it's mitochondrial problem then, and it's coming from mitochondria, then your gene therapy is not helping. And this current treatment, it's vitamin cocktail or specific vitamin, or actually also others, it's not really very good treatment. It's just helping a little bit. And what is interesting, that only some subgroups are sensitive to this vitamin treatment. Some subgroups are not. Good. Thank you. So we can go on. Who will come? So please, don't hesitate. You need more time? Or, okay, if you need more time, then we can wait a little bit, but if you are ready, then please come. So, you are ready. Even if you are not fully ready, it's to be part ready is okay. It's not working.
Hello. Okay. So this is a target drug profile for Alexander's disease. So Alexander's disease is caused by mainly accumulation of GFAP protein in white matter of brain. So we have found a drug ION 373 which is already in the clinical trials, and this is the drug profile for this one. So when this drug was administered, there is reduced in pathological GFP production, and it can be administered for more than 2 years of individual to 65 age, and treatment duration is more than a year, and regimen once in every 12 weeks, and efficacy is showed more than 30% stabilization in gait speed, side effects mild, and therapeutic mobility. As I told, this drug ION 373, so it is an antisense oligonucleotide. It targets specifically GFAP gene, and it silences this GFAP gene, and results have showed reduction in GFAP protein normalization. That's it.
Okay, that is very good, because this antisense therapy is under development currently, but even if it works, then it's not perfect.
Yeah.
The best is to have something which is silencing it more efficiently and which could be used as a single dose therapy. And since there is no treatment available, then we will be very happy if there is only very small treatment effect. It is anyway good. Thank you. Very good. So somebody is ready to continue. Okay, maybe a few minutes break and then we will continue. Okay, it's good to start discussion.
How do I connect?
So you have,
I don't have an HDMI port.
okay,
I have to just be type A.
mm-hmm.
Can be done.
Can we manage without?
Okay. How would I present?
You can just and speak what you have here, because
What if
else
I
to do?
transfer it into a pen drive?
Yes, if it's if you can transfer it, and then we can show it from here. I have also, if you need. Okay, okay. So it's USB
Type A.
No. Should go in. Let's see. Not reading it now.
It's not readable.
Ignore what.
Okay. I'll have to get it in another pen drive maybe.
I had one also here. It's my pen drive. No. So you can also try mine because I know that it could be opened there.
Actually, I also added the causes and symptoms which you told, but
Okay,
are these
so you
necessary?
can, as you like, so it's everything is okay.
So I have this Pelizaeus-Merzbacher disease. It is a rare genetic X-linked disorder, and it affects the males. The reason is that females have two X chromosomes, so even if the gene on one chromosome is defective, the other one can compensate. What happens in this disease is there is a lack of myelin formation, and it affects the CNS. It is associated with problems with the white matter of the brain and the spinal cord, and it is classified as a leukodystrophy, which is the abnormal development of the myelin sheath. The causes, it includes the gene duplication of the PLP1 gene, and in addition to that, misfolding can also take place in other manifestations. This PLP1 gene is responsible for making this proteolipid protein 1 and an isoform, that is DM20. PLP1 is found in the cell membrane of the neurons. This PLP1 is found in CNS, DM20 is found in the nerves that connect the brain to the spinal cord, and both the proteins are found in the cell membrane, as I said. They make up majority of the myelin. The symptoms, there are two kinds of this disease, like one is conatal and the other one is like it takes place later on. It manifests later on, which is the classical type, which is the most common one. In classical type, there is hypotonia, which is the weak muscles, and nystagmus, which is the involuntary movement of the eyes, and there is delayed development of motor skills, such as sitting or grasping objects. Some individuals are able to walk, but their ability to use their muscles slowly degenerates over the time, which is the developmental regression. As the condition worsens, this nystagmus, it usually goes away, but the muscle stiffness, which is the spasticity, and ataxia, which is the problem with the movement and balance, head and neck tremors, which is titubation, and involuntary tensing of the muscles, which is dystonia, and jerking, which is coriform, these manifestations takes place. Whereas in case of conatal, this disease, conatal Pelizaeus-Merzbacher disease, there are much more severe symptoms, and there is a high-pitched breathing caused by obstructed airway, which is stridor, and progressive speech difficulties, dysarthria, severe ataxia, hypotonia, seizures, etc. Talking about the life expectancy, with the conatal PMD, there is death in the early infancy or childhood, whereas in case of classical PMD, which is the most common type, there is variable life expectancy based on the symptomatic treatment of the disease, and with proper medical care. It occurs in 1 in 200,000 males. There are symptomatic treatment drugs available, such as botulinum, and anti-spasticity medicine, which is baclofen, and there are anxiolytics and benzodiazepam, such as Valium and Diastat and lorazepam. So there is this drug which I found out on the internet. This is ION 356. The primary product indication is that it improves and stabilizes the motor dysfunction, which is basically taking place due to the myelin sheath misformation. And the ideal result is that there is reversal of neurological deficits and disease progression, halting in PMD patients. The patient population which it addresses is the children which have confirmed PLP1 gene duplication. All age groups of these patients can be treated with this drug, and the mutation types, they include severe to mild cases. We can administer the drug to them. The treatment duration is around 6 months of treatment to observable clinical benefits, and 1 year of lifelong disease control after limited dosing could be the ideal result. For the regimen part, there is intrathecal administration every few months. Intrathecal means we can administer the drug into the cerebrospinal fluid, and there is infrequent dosing, like semi-annual or annual intrathecal injection. The efficacy, the minimum acceptable result is that it delays the progression and preserves the key motor and cognitive function, which are the main things that get deformed in this particular disease, and it restores the myelin and normalizes neurological function. The side effect includes manageable procedure related to the mild systemic effects. There are no adverse side effects. Safe long-term tolerability is there. And for the therapeutic modality, there is this drug. The mechanism of action is that there is antisense oligonucleotide which reduces the PLP1 expression. So in case of gene duplication, we know that there is going to be enhanced protein production. So what this drug does is that it is basically an oligonucleotide, which is the antisense oligonucleotide, and it goes on and binds to the RNA, and which leads to its degradation. So we have a lower production of the protein. And the targeted mutation adapted PLB1 suppression and normalization of myelination is the end result. So that's it.
Thank you. What you mean exactly with this targeted mutation as an ideal therapeutics? What exactly you mean with that?
So what basically happening is that in this particular disease there is gene duplication that takes place. So when we are administering this drug, which is an antisense oligonucleotide, it goes on and bind to the PLP1 mRNA.
Yes, and
so
you have a less expression?
Yeah, because gene duplication will cause heightened gene expression.
But here you plan to switch off one of the genes, or what you plan with this ideal?
So what we will do is that in the intrathecal administration, when the drug will go and it will bind to the mRNA, so suppose we have 2x expression, so the antisense oligonucleotide will go on and bind to the additional mRNAs that are there, and it will target them for destruction. So we will have a lesser level of expression.
Okay, so this means that if you have a slightly different situation than we had before. So you have here two genes. One is not necessary. So if you will have an antisense, then what it is doing, will it suppress both or one? So it in ideal cases that if the antisense will be specific to one gene only. And the best is that if you could somehow kill this one extra gene. But I don't know whether it is somehow possible.
Yeah, I don't think otherwise they would have developed something, but they have only come up with this antisense oligonucleotide therapy.
Yeah. Okay, thank you.
Thank you.
You can take this. So, who else is ready to continue? So, you are ready? You have a HDMI port in your, or you need a USB or, okay, you have. It was no not so easy. Okay, got it.
Good evening, everyone. So I will be discussing about the spinocerebellar ataxia. So this disease is caused due to degeneration of cerebellum, and its connections primarily driven by mutations in specific genes that cause the abnormal protein function. So what kind of protein is involved in this? Like the protein which formed due to the expanded CAG trinucleotide repeats lead to the production of the protein with abnormally long polyglutamine stretches. This toxic protein can accumulate and disrupt the cellular processes like protein degradation and mitochondrial function and cause cell death, particularly the Purkinje cells present in the cerebellum. So due to this, what symptoms arise? That is gait and balance imbalance, that is movement inability, limb ataxia with tremors, speech changes, oculomotor abnormalities, and cognitive dysfunction. So after this disease symptoms appear, the life expectancy is 10 to 15 years. So there are multiple pathways involved due to this abnormal protein production. So as it is a autosomal dominant disease, so there are many pathways like this protein misfolding. These are the SCA genes, that is spirocerebellar ataxia genes. So there are 40 subtypes of these genes. So there are many pathways, and these pathways involve different kind of genes, and these are their core mechanisms. So if the protein misfolding occurs, there will be the toxic protein aggregation, and its pathological consequence will be the neuronal inclusion and cell death. Like this, there is RNA toxicity, channelopathy, mitochondrial dysfunction, transcriptional dysregulation, impaired status, excitotoxicity, synaptic and axonal transport defects. However, we can't target all these pathways because these pathways have role for the normal physiological function as well. So scientists has targeted this excitotoxicity, which can be inhibited to cure this disease. So there is no FDA approved drug in the market so far for treatment of this disease. However, there is a drug called Reluzole. It's prodrug. It is a prodrug of Reluzole. However, it is already approved for the treatment of ALS. So this Reluzole is currently in phase 3 trial, and it has given positive result, and also the NDA submission has been already filed, and it is planned for the Q4 in 2025 only. So the product properties are like the primary product indication is it demonstrates the neuroprotective activity in neurodegenerative diseases, already demonstrated in Alzheimer's disease, and right now it is in this disease as well. The ideal results are proven efficacy in improving cognitive and motor symptoms in Alzheimer's disease and other neurodegenerative conditions as well. So for the phase clinical trials, the patient population recruited were adults diagnosed with mild to moderate neurodegenerative disorders. The results were the broad patient population including early stage and moderate Alzheimer's, spinocerebellar ataxia, and possible other glutamate related CNS disorders. The treatment duration is it is safe and tolerable for at least 6 months of the continuous use. The long-term safety and sustained efficacy over the year of the administration. So there are certain risks and side effects as well, like mild to moderate gastrointestinal symptom, dizziness or headache. Very minimal side effects with favorable safety comparable to placebo. The therapeutic modalities are like oral small molecule of this drug is, as I already told, that Reluzole, that moderates the glutamatergic transmission by enhancing the glutamate clearance, and its It is optimized glutamate homeostasis with broad neuroprotective and anti-excitotoxic effect without hepatic or CNS toxicity. That's it. Thank you.
Thank you. So this disease is very similar to Huntington disease. It's polyglutamine disease, and this riluzole is used in states against Huntington disease.
Yes.
Why you choose the small molecular drug, not genetic therapy as before?
So, sir, there are more than 40 subtypes of this disease, and for each subtype, we will have to silence a different gene, and that can be very challenging, expensive, and also expensive. So, in such cases, it would be more practical for us to go with a broad treatment pattern. It would be like targeting the excitotoxicity, because this drug, while targeting the excitotoxicity, will reduce the mitochondrial dysfunction. Also, it will reduce the NMD overactivation. It will also enhance the clearation of the protein deposition from the gene lymphatic system also.
Okay, very good. So it makes really sense because you have so many genes leading to this group of diseases or disease that you can't with one or you can't with similar therapy address them all. And another thing is that it's not mutation, it's the polyglutamine, CAG triplets. It's not so easy. It's possible, they are trying, but it's not so easy to find a gene therapy to remove these parts from the gene. So therefore it's what you told, it's very clear that this small molecular drug is more likely to help. But is that drug disease modifying, or is it just symptomatic? What you think?
Sir, it's symptomatic. It gives symptomatic relief only, because to modify the disease, like there should be permanent silencing of that overexpressed gene.
So these proteins are accumulating anyway. They just induce less excitotoxicity.
It will enhance the clearance of the deposited protein, but it cannot actually affect the translation and transcription pattern of it.
Are you sure that it changed the accumulation of protein? Maybe it accumulates similarly, just the bad effects coming from that accumulation are smaller, which is more likely, but who knows? Okay, thank you.
Thank you.
So we have at least 3 more presentations, or I'm counting it wrong. So somebody's ready. Oh, difficult. 3, 4, 4. And 3, we have 3 more, I think. 1, 2, 3, 4. If I'm counting it correctly. There is no need to be absolutely ready, even. Yeah. No, I think you are coming, then somebody's coming from there and somebody's. Okay.
in autosomal recessive neurodegenerative disorder. This mainly caused by the defect in the gene called fraxitine, which is FXN denoted. The cause and symptoms of this disease are including like trouble in walking, fatigue, changes in feeling and slow speech. This mainly affect the motor function of the body directly by affecting the FXN gene. The core problem of this disease is poor muscle control and lack of coordination. The primary symptoms include progressive joint and limb ataxia, leading to wheelchair use over the time. The disease occur in early ages, leading to the adult, which is like 30 to 40. The only drug that has been approved till now is omaxiviloxolone, which is an FDA approved drug, which directly, not exactly directly affecting the FXN gene, but it modulating the NRF pathways, which ultimately leads to the some indication or some modification in the FX gene, which is treating, gives to the symptomatic relief of the disease. The primary product indication is slow the rate and the neurological decline, but the ideal result should be it should halt the disease and restore the motor function of the body. The patient population that primarily includes are adult and adolescent, and the ideal should be the patient population of all ages, including the children and the patient, the old ages. The indication duration of the drug is 48 weeks to 1 year, but the ideal indication duration or treatment duration should be lifelong and durable benefits, as it only right now it's only giving the symptomatic reliefs. The regimen that is currently available for the drug is a daily oral capsule of 150 mg, which is on empty stomachs, but it should not sufficient, which is not causing, which is not treating completely the disease. So the ideal result should be infrequent dosing, weekly or monthly, or there would be some one-time treatment that cure the disease. The efficacy of the drug is it slow the progression, it stabilize the symptoms. The ideal result should be any drug should halt the disease or restore the motor function that has been caused by the defect in the FXN gene. The risk and side effects that are been observed for the drug are elevated liver enzyme, headache, nausea, fatigue, abdominal pain. The ideal side effect or should be infrequent. Because of the infrequent dosing, there should be no side effect as such, but I couldn't find that information, so I didn't put that on the therapeutic modulation that the drug shows, the function or the pathway that it is modulating its NRF2 pathway while it treating the motor function and all that. The ideal therapy or therapeutic application should be an gene therapy, or there should be some direct link or some direct modulation to the FXN gene that can cure the FA disorder. Thank you.
Thank you. So that what is now different is that this disease has accepted drug, this omaveloxolone, but it is really the efficiency is very modest. So if you will find the drug, which is, I don't know how good is efficacy, but it is considered to be modest. If you find the drug, could be another activator of NRF2 pathway, which is stronger, effect is slightly better, then it makes sense to develop that further. If you will have a drug that is as effective than this omaveloxolone, then it is maybe not practical to develop that further if the side effects are only better. What do you think? How the side effects listed here, are they serious? Are they very serious? Not very serious. I mean, if you take just the names, then they are not so serious when you consider how serious is disease. So it's better to have these side effects than disease symptoms. If you have a mild disease, then they are of course serious, but this is serious disease. Okay, thank you. Very good. So we have few more presentations. I will try.
अब इस लैपटॉप का ना मुझे आइडिया नहीं है लेकिन HTML
Good afternoon, everyone. So I'll be presenting target drug profile for Creutzfeldt-Jakob disease. Creutzfeldt-Jakob disease is a rare, fatal neurodegenerative disorder, and it is caused by abnormal folding of prion proteins in the brain, leading to neuronal death and spongiform changes. Spongiform changes are the neurons degeneration, which creates vacuole-like structures and sponges-like structures, so hence they are called spongiform changes. Symptoms include rapidly progressive dementia, myoclonus, ataxia. Myoclonus is involuntary muscle contractions, rapid twitches, ataxia, visual disturbances, and behavioral changes. The disease progresses quickly and the life expectancy is typically less than 1 year after symptom onset. There is no definitive cure. Current treatment is only supportive and it is aimed at relieving symptoms and improving patient discomfort. Product properties, minimal acceptable result and the ideal result include primary product indications, relief of symptoms in prion-induced neurodegenerative diseases. Ideal result would be delay or halt in neurodegeneration caused by the prion infection. The population affected, adults with rapidly progressive dementia and neurological decline are more vulnerable. Adults at early stages of disease or at genetic risk. Treatment duration is chronic. Regimen is supportive therapy and symptomatic treatment is only available. Early diagnosis with targeted anti-prion or neuroprotective therapy can be given. Efficacy of the product property, minimal improvement, median survival 6 to 12 months. As the life expectancy is only 1 year, so survival rate can be increased by 6 to 12 months only. Significant slowing of disease progression, improvement in survival and function. The risks and side effects of the disease, the drug would be rapid neurological deterioration, myoclonus and cognitive decline, devoid of severe, the ideal result should be devoid of severe CNS deterioration and secondary infections from the targeted drug. Therapeutic modality, supportive and palliative care can be given, and the disease modifying prion targeted therapy can be prion targeted therapy can be given. Thank you.
Thank you. If you can just go back to this table. And there is one thing is that, so this is the side effect of your drug. So it's, if your drug has a side effect that is rapid neurological deterioration, so this means that it's very, how to say, toxic drug. So it's not probably what you mean with that. So that usually the side effects should not be too strong. If side effects are so strong, then it's too much. In ideal case, the side effects should be very, very mild, and there's minimum acceptable side effects can't be very strong. They can't be worse than disease itself. Okay, thank you.
Yeah, okay.
Thank you.
So I will try again. It is coming. No. What? So cancel.
Close connection. एक ही है वही यूएसबी पोर्ट
Or you can put it to memory stick also and to my computer if necessary. Have a memory stick. It's easiest to copy as we did earlier.
Yes, ma'am.
Or you have another laptop. So
डिस्प्ले नहीं आ रहा हमको भी
Okay, so I'll be talking about X-linked adrenoleukodystrophy. So it is a rare inherited peroxisomal disorder which is caused due to the mutation in the ABCD1 gene. It leads to the accumulation of very long chain fatty acids in the brain, spinal cord and adrenal cortex. So the major symptoms of this disease is adrenal insufficiency, neuropathy, ataxia, loss of vision, hearing and speech and other cognitive disabilities. So the protein crucial for transporting the very long chain fatty acids, which is the XQ28 gene, it is crucial for transporting the long chain fatty acids to the peroxisome. It is broken down. So for the treatment of this disease, FDA had approved Skysona in August 2021. Its chemical name is allyl diodogene autotemcel. So the review of this SkySona gene therapy, it was done with the help of lentiviral vectors. It was used with the help, so the lentiviral vectors, they helped in transporting hematopoietic stem cells and inserting into the ABCD1 gene through the lentiviral vector therapy. So there was this link which is not opening right now. It was from the official FDA website. It's not opening, but it majorly suggests about the dosage. The dosage of this therapy is 5 into 10 raised to power 6 CD34 cells per kg according to the body weight. And this study which was done by FDA was done majorly on the male population. 26 males were recruited, and the major warnings or the major limitations of this therapy was hematological malignancies, including life-threatening cases of acute myeloid leukemia having occurred with the patients treated with SkySona. So coming back,
as she said that the primary goal of SkySona treatment is to reduce the rapid expression of neurological deterioration that is associated with cerebral ALD, that measured primarily by the MFD, that is major functional disability free survival. And this is found out that out of 26 subjects, 23 achieved MFD free survival at month 24, exceeding the 50% clinical benchmark, that is 88% of MFD free survival, and achieved 92% resolution of cerebral inflammation. And for patients who had already become symptomatic, then this SkySona treatment provided a significant protective effect compared to the untreated natural history cohort. And there is a significant reduction, which I previously discussed, with this major disability functional index. And there is a symptom stabilization and neurological functional improvement with this therapy. That is, there is a sustained neurologic functional score for at least 24 months post-treatment that is experienced a period of NFS stability. And this therapy operates by slowing the rate of neurological deterioration rather than offering reversal of pre-existing symptoms. And the therapeutic dose that ensure the sufficient engraftment of the modified C34 positive cells to establish sustained ALDP expression, that is 12 into 10 to the power 6 CD34 positive cell. And for this, the autologous stem cell transplant that involve harvesting the patient's own CD34 positive HSC, and these cells are further genetically modified ex vivo to include a functional copy of this ABCD1 gene, and then the modified cells are then reinfused, establish a long-term source of ALDP expression cells. But there are some adverse events that are happening with this SkySona therapy, that is the hematological malignancy. That is, 3 subjects were identified with a subsequent diagnosis of hematological malignancy following treatment with SkySona. And the 2 subjects enrolled in this clinical study died following treatment, emphasizing the need for continuous post-treatment safety surveillance. And there are other common adverse effects that is associated with post-SkySona treatment, that are febrile neutropenia, that is low WBC count, mucositis, that is there is an ulceration in the mucous membrane, and the Sizer cases prevalence in 7% of the population. And there is a clinical success with this SkySona, that is provide a comprehensive durable treatment effect that preserve the quality of life for the patient. That first of all is neurological stability, sustained gene expression, reduce very long chain fatty acids, and preserve the quality of life. and the minimum acceptable therapeutic results are, that is, the patient must remain free from MFT within critical 24-month period post-treatment, and the measurable and detectable expression of the ALDP protein that confirming successful gene transfer and engraftment. Thanks.
Thank you. So you've spent a lot of time to do it. So actually I have no questions, so it was clear. Only one thing what you mentioned, that you said that gene therapy has side effects. So it could indeed happen, because if you introduce the gene back, you don't correct mutation, but you introduce the gene back or something, then you will put many copies back, and you can have side effects because you have too many copies, or for some other reasons. I'm just adding the side effects. Yeah, so thank you. Yes. Do we have one more presentation? So, or I'm wrong? Okay, so it's done. Good. So then, what do we conclude? So it was relatively random list of diseases, rare diseases having neurodegeneration or neurodegenerative component. And with only one exception, they were all genetic diseases. So what is the best treatment option for genetic diseases? As you saw, many had already the treatments, gene therapy treatments, or at least if they had not yet, then you propose that this is good treatment. So actually for rare genetic disease, the best treatment is to correct the mistake or mutation. So a lot of new gene therapies are coming to market. What is the problem with them at the moment? The price. Usual price for this one treatment is $2-3 million. So means that in US they can do it. In Europe it's already expensive, and in other countries it's too expensive. It takes time. Maybe it is taking a long time, but after 20 years at least, it will be cheap. Because what is expensive? Expensive was to develop this, but the price itself to produce this therapy is not expensive. So it is best ways to correct mutation. It's now expensive, in future it will be not. Then another thing you showed, that there were therapies, oligosenses, to suppress expression. So this has limitations, because you can use this oligosense suppression only if there is gain of function mutation. You produce something too much, or it's doing something wrong, and then you suppress it. But if it's lack of the expression, then it will not help you. This is one limitation, and another limitation is that you have to treat with them all the time. So it's cost. So sometimes it works, but it's not as perfect as correction. But you should correct it quickly, because as you remember, the symptoms are strong, frequently starting very early. Damage is coming quickly. You need quick diagnosis and quick correction. So this is, I think, the future. And sometimes this is not possible, gene therapy. When it's not possible, if disease is absolutely very rare, who is developing this gene therapy? If there is, as there was in case of spinocerebellar ataxias, a lot of genes involved, a lot of mutations involved. So it's difficult, you can't target them all, although they will try to do that. And some mutations are not easily correctable. If you have a mitochondrial mutation, you can't correct it nowadays. Also, the GAG repeats maybe are more complicated to correct. And then you may have unexpected side effects. Sometimes, in last case, they were quite serious side effects. So sometimes you can't avoid them. So then the solution is these small molecular drugs. But this is for genetic diseases. They are not helping you in case of Alzheimer, Parkinson, ALS. Okay, so this was the summary. So just few minutes and then we will continue with lecture. So if it's 5 minutes, not more, otherwise we will finish too late. Okay, so 5 minutes.
Easier, quick 5 minutes break. Walk, take your grab and finish and come back.
Just there is one good book. I got part of my knowledge from this book. It's a practical guide to drug development in academia from Spark. Spark is the funding organization funding this translational research. So that this is, if you search from the web, then you really will get a lot of rubbish and not so easy to find. So when I prepared my presentation, so I got it mostly from this book, and also I attended one course they organized. Because if you are biologist like me, then you really don't know these things. You need some education. It's okay to stop. It's okay if we will go a little bit past 5 also. If we will finish a little bit after 5, because I don't know how long it goes. Okay, okay. So the mitochondria and neurodegenerative diseases. So I will speak about the mitochondria, and I will try to include diseases to this mitochondrial function I'm explaining. So if you take the history, then first this initial introduction of the concept of mitochondrial disease was by Robert Luft, 1962. And then what was most relevant after that, they discovered that part of the mitochondrial diseases are because of mutations in nuclear DNA encoding mitochondrial genes. Then respiration rescue by mitochondrial transfer, 2006. Somebody from you was studying it. She's not here at the moment. And then now, a few years ago, first FDA-approved drug, Omaveloxone, that indirectly at least affects mitochondria. So mitochondria, as you may know, they are actually bacteria. So long time ago, this giga-year is billion years ago, few giga-years ago, the bacteria, Rickettsiales, it's double membrane bacteria, fused with prokaryote, which we have now calling prokaryotes. And after that, our cell contained these bacteria. So there is, at the beginning it was simple symbiosis, now it's something more complicated. So the mitochondria, you see the structure, there is outer membrane, then there is inner membrane that has a lot of such kind of foldings, where you have respiratory chains and other such kind of things. Between membranes you have the intermembrane space, and then inside you have mitochondrial matrix, where you have everything from mitochondrial DNA to ribosomes to produce mitochondria encoded proteins. So and these foldings, they are called mitochondrial cristae. So the mitochondrial DNA and nuclear DNA. So the mitochondrial DNA encodes 37 genes, and nuclear DNA encodes much more genes. A part of these are going to mitochondria. How much exactly, who knows, but hundreds or maybe 1,000. If you take the mitocarta, this is the list of mitochondrial proteins, then I think there are few thousand proteins in that list. So most of the mitochondrial proteins that are in the mitochondria or attached to mitochondria are coming from nucleus, and very small part are coming from the mitochondrial DNA. And you have in every mitochondria, you have many copies of these mitochondrial DNA. I don't know what exactly is the average, 10 copies or something. There are such kind of circles there. So mitochondrial diseases, how relevant they are. So it's difficult to tell. First, there are primary mitochondrial diseases, which are clear, usually genetic, inborn genetic diseases that are affecting primarily the mitochondrial function. So how much it's expected that altogether these diseases, it's 4,000 individuals, I think per million if I'm correct, have a primary mitochondrial disease. So these diseases are caused by mutations in mitochondrial DNA and in nuclear DNA. Then there are secondary mitochondrial diseases that are not really related to these specific mutations, but they are acquired or they are accompanying other diseases. So all these neurodegenerative diseases, you remember, they had some mitochondrial component. How prevalent are these secondary mitochondrial diseases? It's not possible to tell, because it's not possible to tell what is the primary reason, what is the secondary reason, and what is the role of mitochondria. But some people claim that they are very frequent. For example, one study says that maybe 80% of children with autism spectrum disorder have some kind of mitochondrial dysfunction present. So it's very variable, it's not possible to tell how frequent. So all these diseases almost we had are secondary mitochondrial diseases. What is missing here, what is clearly missing is MS, but it also has a very small mitochondrial basis. So I tried to find out somehow how strong is the mitochondrial component in development or in progression of these diseases, because even if mitochondria are not the primary related to primary defect, they are the reason why disease is progressing. There is such kind of cycling progression. So I asked from yesterday from ChatGPT what is the mitochondrial contribution. So as you see, it's quite big in case of Parkinson's disease, less in Alzheimer, less in Huntington, less in ALS. And also if you take the psychiatric disorder, they have the mitochondrial component. At least ChatGPT proposes that they have. Because we don't know much about the neuropsychiatric disorders, it's more difficult to tell how important the mitochondria are there. In addition to neurodegenerative diseases, mitochondria are playing the main role in many, many different diseases not related to brain or peripheral neuronal system. So we will continue with mitochondrial function. What do mitochondria do for the cell? Why cells and neurons specifically, why they need mitochondria? So it's many things. What is very well known that they produce ATP, 65 kg of ATP per day as an average. It's quite a lot. Then the mitochondria are death signal indicators or inducers. So they regulate apoptosis. They buffer calcium, they produce ROS, they produce different kind of things. They are thermogenetic. So we don't have a brown adipose tissue, but mice have, and mice use mitochondria to produce, heat if it's highly necessary. Mitochondria are relevant in immunity, and they are metabolic hub as well. So let's cover now the main things. So mitochondrial energy production. So the food is going in and the energy is going out. Food is what mitochondria will take in to produce energy. It is pyruvate. Mostly it's pyruvate. Neuronal mitochondria at least use the pyruvate. Pyruvate is coming from glucose. So we have in cytosol glucolysis, and pyruvate is a product. Pyruvate is going to mitochondrial matrix. So it uses for that mitochondrial pyruvate carrier that is in mitochondrial inner membrane. So if mitochondrial pyruvate carrier is deficient, then the mitochondria do not have any more food, they can't produce energy. So this leads to what? If pyruvate is not going in, then more pyruvate is going to lactate. So accumulation of lactate. So it's rare genetic disease, 1 to million, mitochondrial pyruvate carrier deficiency. It leads to lack of energy, lactic acidosis, too much lactate, poor growth, neurological symptoms, and so on, because there is no energy and too much lactate. So if pyruvate is already inside of the matrix, then there is enzyme pyruvate dehydrogenase complex. But not only, so you see there is two enzymes. Actually it's two ways. It is possible that it will be going to oxaloacetate, but if you do not have enough energy, then pyruvate dehydrogenase is active and it will go to acetyl-CoA. So this enzyme is itself multi-enzyme. It has 3 catalytic components, and not going to details. So this pyruvate will be going to acetyl coenzyme A, and the reaction is giving you one NADH molecule as well. So pyruvate dehydrogenase complex deficiency. Many different mutations in this complex. It's quite prevalent, 1 to 40,000 in North America, and again, lot of different kind of symptoms, abnormalities in the brain, mental retardation, and so on. Not enough energy. Then you have this acetyl coenzyme A, and this is then combining with citrate and is going to citric acid cycle or Krebs cycle or 3-carboxylic acid cycle or just TCA cycle, different names. So again, not explaining what kind of enzymes you have here, but what this means that all these enzymes remove carbon and capture the high energy electrons. They will get to produce NADH and little bit of, where we have it, little bit, we don't have that in figure, I can find it, also little bit of FADH, so nicotinamide adenine nucleotide and flavin adenine nucleotide. So, and it will end up here, and so point is that it produces a lot of NADH and also a little bit of FADH and ATP. So the Krebs cycle disorders, if something is wrong with this enzymes in this cycle, succinate dehydrogenase deficiency, aconitase deficiency, alpha-ketoglutarate dehydrogenase deficiency, and fumarase deficiency that I have here as an example. So if you don't have, it's in this place, if we don't have a fumarase, then we have as a result too much fumarate accumulating and not enough malate. So it's very rare. It's as it says, it's 13 cases and now 20 cases more in the world, and symptoms are again quite serious and similar. So, but if this cycle is working, then you have a lot of NADH. What the cell is doing this with this NADH? It uses it in mitochondrial electron respiratory chain. These are enzymes in the mitochondrial inner membrane. Complex is 1, 2, 3, 4. So these complexes are multi-protein complexes, a lot of subunits. And in addition to these membrane complexes, there are ubiquinone and cytochrome C that are mobile carriers of electrons from one complex to another. So again, not going to details, what happens is that these complexes will take NADH and they will pump protons to intermembrane space. So you have proton gradients. So these protons will come back through the ATP synthase, which makes ATP when the protons will come back to the matrix. So you have a lot of ATP, and you have a lot of diseases related to this mitochondrial respiratory chain complexes. So some were already covered by you when you made presentation. Maybe two most important diseases. It's Leber hereditary optic neuropathy, which is related to mutations in complex 1 subunits. Complex 1 ND1, it's not the hydrogenase subunit 164. So it's most common, 1 to 30,000, 1 to 50,000. And the main symptom is that there is optic neuropathy. You will lose your sight. Then another symptom which was covered was this late syndrome. Mutations in the mitochondrial DNA and in nuclear genes which are involved in cytochrome C oxidase. So 1 to 40,000 again, quite a lot, and in some places, Faroe Islands, even much more. This disease is more serious. It's the life expectancy is very short. So then you have your ATP produced, but your ATP is between the mitochondrial membrane. So, sorry, your ATP is in mitochondrial matrix. So I'm becoming tired and doing mistakes. So you have your ATP here. The ATP should go out somehow. It's required to in cytosol. So for that there is adenine nucleotide translocase. So the adenine nucleotide translocase is sending ATP out and taking ADP in. It's in the inner membrane. And it's also, if it's mutated or if it's deficient, then leads to disease. It leads to Sanger syndrome. So there are two possibilities. One possibility, which is very rare, is that you have mutations in the translocase itself. Another possibility that you have it in this RGK gene encoding the kinase, which is required for the export, which is required for the, how to say, functioning of this translocase. So again, different symptoms, 40 cases reported worldwide. So if you have your ATP, then usually one molecule, from one molecule of glucose, you will get 32 molecules of ATP. If something happens, one of these diseases, or if mitochondria are not fully happy, then you will have less ATP. So it is mitochondria and energy production. Then mitochondria and cell death. So the cytochrome C, which is in intermembrane space transporting electrons between respiratory chain complexes. This could go out from mitochondria for some reason. If mitochondrial outer membrane becomes permeable to it, there is mitochondrial outer membrane permeabilization. Frequently people use just MOMP. If this MOMP happens, then the cytochrome C will go out. It's bad. Why it's bad? First is that you don't have any more your electron transporter there. Mitochondria can't produce ATP after that properly. But what is another thing is that it will activate Gas bases. And as you know, Gas bases will lead to apoptosis. And it's not just this cytochrome C who will sneak out. It's also the SMAC and Diablo. SMAC is second mitochondria-derived activator of Gas base, and Diablo is direct inhibitor of apoptosis-binding protein with low isoelectric point. So this SMAC and Diablo will also help to initiate apoptosis. It's not clear how this MOMP happens. So people even don't know whether this apoptosis spore really even exists and how it looks like. But what is maybe consensus that there is PAX protein. PAX protein is usually doing nothing. But if the pH3 only protein will interact with this PAX protein, then the PAX protein will form the pore, which is MOMP pore. And cytochrome C, SMAC and Diablo will sneak out. It could also lead to megapore, or maybe, and in that case also bigger components will go out. How this happens, how this pore is formed, so you need this pH3 domain here. So you have pH3 activators which will activate this PAC. Or you have pH3 mimetics that block the BCL2 proteins, and BCL2 proteins do not anymore inhibit PAC after that. So your PAC is becoming active and induce MOMP. Or you can have a PAC inhibitors to block the MOMP. So it's mostly for the cancer therapy. So in case of cancer therapy, it's necessary to kill the cell. So these pH3 mimetics and activators, they will induce MOMP and kill the cell through apoptosis. So the venetoclax is the one which is in the market currently against leukemias, some form of leukemias. So theoretically, what is the problem? We have too much MOMP sometimes in neurodegenerative diseases, and these inhibitors of this MOMP could be therefore protective, theoretically, but nobody is in clinical trials because it's too risky. If you block this important pathway here, you can expect huge number of problems. So this is mitochondria and apoptosis. Then you have a mitochondria and calcium. So I was looking yesterday for good calcium homeostasis image in neurons, but it was really difficult to find. So it is such kind of simplified image, how intracellular calcium fluxes are happening in neurons. If your membrane depolarizes or some other channels open, there are quite many channels in plasma membrane, then calcium will enter to the cytosol of neuron. Then what it is doing after that? Or is another option is that you have intracellular calcium release from endoplasmic reticulum. This opens the channels in the plasma membranes, and more calcium will come in. So after that, this means you have a lot of calcium in cytosol. This calcium will be taken up to endoplasmic reticulum by SERCA, and then later it will be again released by IP3 receptor or ryanodine receptor. And then people are showing there is mitochondria have uniporter, which takes the calcium in or lets it out if concentration is low outside. It's a simplified figure. It's much more complex because these mitochondrial and calcium and ER calcium signaling are happening together. So mitochondria are very close to endoplasmic reticulum. They are forming such kind of mitochondrial contact sites. And in these contact sites, where you have a physical contact, really, protein-protein contact between ER and mitochondrial proteins, the calcium released through IP3 receptor from ER is going directly to mitochondria. Contact is so tight that it's not leaking out, it goes from one organelle to another organelle. It's relevant because mitochondria can buffer calcium better. And it's also relevant that mitochondria need it because the calcium or these calcium spikes, they will activate the mitochondrial respiratory chain. Many mitochondrial respiratory chain dehydrogenases are calcium sensitive. Pyruvate hydrogenase wants calcium to work. So this is relevant for cell and for mitochondria to have these kind of contacts. And it's not only calcium that moves through these contacts, but we are currently speaking about calcium. Why these contacts are relevant in case of neurodegenerative diseases? The reason is that these contacts are very complex. It's not just IP3 receptor and some channel in mitochondria. You have a lot of different proteins in these contacts, and these proteins are quite close to diseases we had. If we take the Parkinson disease, TG1, sigma 1 receptor, VDAC, pink-parkin, mitofusin, they are all in these contacts. Mitofusin is one of the proteins helping to form these contacts. If we take the amyotrophic lateral sclerosis, then again many proteins involved there or mutated, they are very close to the complex or in the complex. Or we have rare Wolfram syndrome we are studying. Again, everything is in that complex. So people are now quite sure that this complex is playing important role in neurodegeneration. If you have too good contact, then maybe calcium fluxes are too strong. If you have bad contacts, then calcium is not going to mitochondria. It is maybe not primary reason, but it's one of the factors contributing to neurodegenerative disease progression. And it's in many cases present, not just one rare disease. Then mitochondria and ROS production. So we will go back to the same respiratory chain figure. So usually what the figure you have in textbook is that NADH is going in and protons are coming out and ATP is produced. But you have a lot of electrons who will sneak away from this pathway. And these electrodes are they are reacting with oxygen, and they are forming superoxide and hydroxyl peroxide. First superoxide, and then you will get hydrogen peroxide after superoxide dismutase converts it to superoxide to hydrogen peroxide. And this is how you will get the ROS. If you you always have a ROS, so it's small fraction percent or so is always 1% or so electrons will always sneak away and divert it to ROS formation. If it's so low, then it's okay. Low level ROS is always good for something. First it's good for something, and then it's good for the different kind of reactions. It's necessary to activate something in case of hypoxia, immune activation, inflammation. So if you completely block it, no ROS at all, then it's not good as well. If you have too much ROS, then you have excessive oxidation of lipids, protein, DNA. This is clearly damage. If you have too much ROS, then what cell is doing and what are the options? So that is what for the cell has antioxidants. So first there are enzymatic antioxidants starting from superoxide dismutase, and then catalase, glutathione peroxidase, glutathione reductase, who will all remove these free radicals, not going to reactions now. And then you have non-enzymatic antioxidants, endogenous and exogenous, different vitamins, melatonin, whatever, who will, most importantly, glutathione, coenzyme Q, vitamins, who will help to reduce the proteins which are oxidated. So if you have too much of ROS, then you need antioxidant theoretically. So there is one very popular antioxidant, MitoQ, mitoquinone mesylate, synthetic analog of coenzyme Q10. So the logic was that if ROS are produced by mitochondria, then most effective antioxidant is the antioxidant which goes to mitochondria. So they made a construct which is going to mitochondria and should be protective there. So it's very old story already, and at the beginning they were quite sure that it works well. So, but it was not working. It was, I think, 15 years ago when they checked whether the MitoQ helps in case of Parkinson's disease, and it was not really helping. After that, I checked yesterday this clinical studies trials database, US clinical trials database. It has not been tested there in case of neurodegenerative diseases, but there are many currently running clinical studies, MitoQ and some other diseases. So they still hope that there are some where when it works, but no hope with neurodegeneration. What is the reason? No idea. Maybe whether it's not effective or whether it's not relevant to block the ROS in mitochondria. Then there is another way also, that there is oxidative response pathways. When these antioxidants and antioxidant enzymes are not strong enough, you still have oxidative stress. Then cell is activating the oxidative stress response pathways. The most well-known is SCAP1 NRF2 pathway. You had that in one of the diseases and the drug affecting this pathway. So the logic is that if there is no oxidative stress, then the protein named GAP is interacting with protein NRF, and NRF will be ubiquitinated and degraded. If you have a lot of ROS, then this ROS will block GAP1. You have a NRF free to do whatever. It goes nucleus and it will activate their expression of antioxidant proteins. So it's work against oxidative stress. So if you take the ILS, then the first gene discovered was SOD, superoxide dismutase, which is in mitochondrial intermembrane space and in cytosol. So it's clear that ROS, at least in this specific case, the ROS is the reason why ILS starts. And it's logical to use here the antioxidants or theoretically the pathways which activate the antioxidant response. And if you take again the clinical trials for ILS, then there are many such kind of antioxidants or things which affect oxidation and other things, ER stress, mitochondrial stress, and so on. Okay, then mitochondria are doing a lot of different synthesis processes or doing a lot of synthesis. I have here as an example only iron. So it's not so well known maybe, but it's again very relevant. Iron goes to mitochondria, and iron is used in mitochondria to produce ferrum sulfur clusters or Hem. Okay, we don't really speak too much about the Hem, but we speak about the ferrum sulfur clusters. So there is the pathway, complex pathway to produce these clusters. These clusters will then go back to cytosol, and they are relevant there because many cytosolic enzymes require these ferrum sulfur clusters to be active. But one of the enzymes or proteins involved in this process is at the beginning is frataxin. So this frataxin is required for the formation of the two ferrum to sulfur clusters, and then when they are formed, then they will go on through this pathway. So if you don't have this frataxin, what happens? You anymore can't make these clusters. It's bad that you don't have these clusters anymore in cell, but it's also bad because the iron will be not used and the iron will accumulate in mitochondria. Too much iron in mitochondria, it's toxic. You have free radicals, oxidative damage, no ATP production. It will kill the cell. So Friedreich ataxia, you had it before, you heard the symptoms, serious symptoms. And the drug, omaveloxone, is inducing the NRF activation. For some reason, this oxidative stress you will get from iron accumulation is not activating the NRF properly. And you need this NRF activator, which helps then to block this oxidative stress you will get from this iron accumulation. Not only iron accumulation, but this is one of the reasons. And this is just to show how effective it is. Placebo is black line, disease progresses. Blue is the omaveloxolone, and there is clear protection, although the progression happens also here. So all these drugs which are coming and used, they are, how to say, not just from out of blue. They are designed specifically against the mechanisms of these diseases. And then another thing, still some minutes left. One thing is that mitochondria is doing a lot of things that are required for the cell. But mitochondria must be healthy to do everything. So there are a lot of systems which keep mitochondria healthy. And one of the things we are studying is mitochondrial dynamics. So you need that mitochondrial dynamics to keep mitochondria happy. So this mitochondrial dynamics means that mitochondria are moving. There is mitochondrial traffic. Mitochondria are fusing with each other and then breaking apart, fission. And most importantly, there is this mitophagy, mitochondrial quality control. So I have one video I'm always showing. Just to explain how complicated is the mitochondrial dynamics. We'll see, will it come? That's not coming like So when we started to study mitochondrial dynamics, then it was only mitochondrial trafficking. So mitochondria are moving through the axon anterogradely, retrogradely. Then after that, mitochondrial fusion and fission was, how to say, discovered and started to be popular. As the system became more complicated and complicated. Why I'm showing it, it is like the traffic you have here. It's quite close. But okay, so the point is that this mitochondrial dynamics, movement, fusion and fission and mitophagy, they are three components which keep the mitochondria happy. So why this is necessary for mitochondria? First, if you take the neurons, then they move, in neurons, mitochondria are moving from one place to another place. It's relatively random movement, but there is system in it. You have the fresh, good mitochondria coming from the body, and these fresh, good mitochondria are going to the end of axon. They will become relatively old there. If their axon is 1 meter long, then this mitochondria is not anymore having new components. It should go back to body to get new components. This was the one point. And another point is that in neuron, in axon, you need energy in different places at different moments. You need energy in synapses, in some synapses. So where there is energy loss, mitochondria will go to this place to provide energy there. So this is, I think, quite well shown nowadays. So mitochondria move all the time. So just checking a little bit. So this is axon. Axon is starting from here, cell body, it's branching here, branches go like that. And there is sometimes sometimes they are moving. So this is to send mitochondria to places where they are required. Old mitochondria, they can come back maybe to body and so on. And there are kinesinopathies. So this process, as you saw, requires kinesin for anterograde transport and thymine for retrograde transport. So the kinesins could be in trouble. You have mutations. So if your kinesins are not effective, you may get some disease. So you have a lot of kinesins. They are related to hereditary spastic paraplegia and Charcot-Marie-Tooth disease type 2. They are not leading, I mean, they are small part of causes, so it's not a major player. They're not the major players. Then there is mitochondrial fusion and fission that happens all the time. So this requires specific proteins. Mitofusin to fuse outer membrane, OPA1 to fuse the inner membrane. So they are also related to diseases. So, but before going to diseases, I saw the process where red mitochondria is fusing with green mitochondria. So they fused. Red and green become together, and now they will just depart. They will break to pieces again. So we use this method to quantify fusion and fissions. But again, if these proteins, fusion proteins, are deficient, mitochondria do not fuse anymore, they become fragmented. They don't produce enough ATP, then this leads to disease. This OPA1 deficiency is quite frequent reason for the autosomal dominant optic atrophy. Frequent means that you see 1 to 12,000, 1 to 50,000. It's most common hereditary optic neuropathy actually. Supportive treatment only. Or this mitofusin mutation is main reason for the CM2A, if I'm correct. So this means that if mitochondria can't move, can't fuse properly, they're not happy, and there is some kind of disease coming after that. And then at the end, the last slide is about the mitophagy, Pink and Parkin. This I already told you before. Thank you. Do you have some questions at the end? So I tried to give you quickly the overview about the mitochondrial function. And also, I hope this explained you how many of diseases you had before, how the treatment of diseases is related to the mechanisms. That if you usually are, how to say, checking what the drug is doing, then we are, how the drug is used for the treatment, and we are not knowing exactly what it is doing or not understanding how it's involved in this disease mechanisms. But here all the drugs we covered Monday, Tuesday, I think we had all these drugs now in these mechanism lectures. So any questions? Okay, then we will have to finish today. Thank you very much. Yes, please. And after that, they will divide, and it is the figure we have red and green mitochondria. We use the laser and specific probe to make one mitochondria. They were green, we made one mitochondria red. And if we wait, for example, 1 hour, then we don't have this red mitochondria anymore. This red will be everywhere in other mitochondria. Insufficient means that we will do red. Okay? And that is all presentation. Okay then, till tomorrow. Okay.